We have a few housekeeping notes to be reviewing while we get started. So please take a moment to introduce yourself in the chat in Zoom or on YouTube. Tell us your name, your affiliation, or where you're calling in from today, and what are you excited to learn about? Use that chat button in Zoom and make sure you're sending your message to all panelists and attendees to introduce yourself, or just use the chat on YouTube. To ask questions from our NGL, Next Generation Leaders speakers, please use the Q&A in Zoom. You'll find that button down in the footer of your Zoom window. It says Q&A. Make sure your questions end up there, not in chat, so that we make sure we see them. And make sure to include the speaker's name if the question isn't about a specific presentation. But for those Allen Institute questions or presentations, those are pre-recorded. So we're not going to have live Q&A during the event. Please visit community.brain-map.org/tag/showcase2020. Or if you go to that community.brain-map.org, you'll see it on the main page for a group of threads on those project talks to ask questions of those teams. And they'll be keeping an eye on those threads and answer them. Allen Institute staff and NGLs, we have our private Q&A and roundtables. You should have the links for that. If you're struggling to get connected on Zoom, you have a couple of options. You can view this event on YouTube. Go ahead and click that live button in the top corner of Zoom to get yourself bumped over to YouTube, or back on the login page where you signed into Zoom for this event, there is a direct link to YouTube, or try switching to phone audio to minimize the strain on your bandwidth. Here's today's agenda. You can also find that on the registration page for this event. We're not going to have breaks today because it's being live streamed and recorded on YouTube, so you can catch up at any time if you need to step away. Good morning, everybody. Thank you for joining us today at the Allen Institute Virtual Showcase Symposium 2020. We are going to get started at the top of the hour. We have a couple of housekeeping items to go over in the meantime. First of all, use that chat panel here in Zoom or the live chat on YouTube to introduce yourself. Tell us your name, your affiliation, and where you're calling in from, and what you're excited to learn about today. That's that chat button down in the footer of Zoom. And please make sure you're sending those chat messages to all panelists and attendees so everyone can see them. If you have questions for our next generation leaders who are speaking today, please go ahead and ask those using the Q&A panel. You'll find that button down in the footer of your Zoom window as well. Please make sure to include the speaker's name in the question if it's about a specific presentation so they know which questions are for them. But we won't have live Q&A for those pre-recorded Allen Institute project talks. We do have threads open on our community forum at community.brain-map.org for you to submit questions to those talk teams, and they'll answer those. They'll be keeping an eye on those threads. So if you go to community.brain-map.org and click on, and you'll find a list of those threads on the main page, or click on the tag Showcase 2020 to get to all of them. Good morning, Daniel Felleman. Thanks for joining us today. Anybody else wants to introduce yourself, please use that chat panel. If you're struggling to get connected on Zoom, you have a couple of options for troubleshooting. You can view this event via the live stream on YouTube. In the top left corner where it says custom live stream service, click on that and then view stream there. That'll send you over to YouTube. Or you can go back to the sign in page for this event where you got this Zoom link and click the YouTube direct link there. Or you can try switching to phone audio, which will minimize the strain on your internet bandwidth. Here is our agenda for the day. We have 3 project talks and 2 next generation leader talks. This agenda is on the registration page that you used to sign up for this event. We won't have breaks during the program today because it is being live streamed and recorded on YouTube, so you can just catch up later. And this recording will continue to be available after today. We will be getting started right at the top of the hour. Thank you for joining us at our virtual Showcase Symposium today, here on Zoom or on YouTube. We have a few housekeeping items to cover before we get started with our presentations. Please join the conversation. Introduce yourself in the chat on Zoom or on YouTube. Tell us your name, your affiliation, and what you're excited to be learning about today. And please make sure to send your chat messages to all panelists and attendees so that everyone can see your message. I see we have a couple of introductions on YouTube as well. Thank you for joining us over there too. But if you have any questions, please make sure that those end up in the Q&A on Zoom, not in the chat panel, so that our panelists make sure to see them. Please make sure to include the speaker's name if the question is about a specific presentation so that they know that question is for them. If you're on YouTube, we will be taking questions from there as well. But for those Allen Institute talks, we are not going to have a live Q&A, so please direct those questions to our community forum. The link has just been put in the chat, community.brain-map.org. Those threads are all going to be on that home page of the community forum right now. So please make sure to post your questions there, and those project talk teams are going to be keeping an eye out and answering those questions. If you're struggling to get connected on Zoom, you can connect a couple of different ways. In Zoom, if you click on Live on Custom Live Streaming Service and then select View Stream, that will take you over to our broadcast on YouTube. It's the same content, just in a different location. The login page that you use to get into this Zoom webinar today also has a link directly to YouTube. Or if you just go to YouTube and search for Allen Institute and click on our channel, you'll find it there as well. Or here in Zoom, you can also try switching to phone audio to minimize your strain on your internet. So here is today's agenda, and it is now time to turn it over to opening remarks by Jenny Close, the chair of the Showcase Committee. You can find this agenda at any time on the event page on our website. We're not going to have breaks today. It's going to be live streamed and recorded, so you can catch up later if you need to. All right, Jenny.
Thank you, Caitlin. Welcome everyone to our annual, now virtual Showcase Symposium. My name is Jenny Close, and I'm a senior scientist on the MFISH team at the Allen Institute for Brain Science. It's been a particularly trying year, but we're thankful to host our 7th annual Showcase Symposium virtually to give an update on our progress and accomplishments this year and to hear from our excellent new cohort of next generation leaders. I'd like to take a quick moment though to thank the Showcase Committee, our group of internal volunteers who spent a lot of time helping organize this program. I'd really like to thank Stephanie Seaman for working with project talk teams with me and developing the schedule, and Caitlin Cosimo and Ashley Bachman for recording the 9 talks. That was really convenient and amazing to be able to pre-record everything. I'd like to thank Saskia DeVries for organizing the roundtable discussions, Christina Jarvis for prepping our next gen leaders, and Sarah Naylor and Jennifer Pulaski for their support. I'd also like to thank the teams across the Institute, both scientific and administrative. Whether you are contributing a project talk today or not, I know everybody's working hard over the last year. We've all been flexible and working from home or kept things moving at the Institute as phase 1 and 2 employees during our ongoing modified work lives due to COVID-19. I'd like to thank the animal care technicians, finance, IT, and legal for keeping everything running, especially the RAs, scientists and investigators. Team science has truly been tested this year, and we're grateful for everyone's contributions to continue working toward our mission. Thank you so much. Today and tomorrow, we will also introduce you to the new class of next gen leaders. Next slide. These 6 individuals were selected by a large internal selection committee from a highly competitive international pool of applicants to serve as peer advisors for our junior scientists. They are rising leaders in the field, and you'll have the chance to hear presentations on their current research. Our new NGLs are Clinton Cave, assistant professor at Middlebury College, Lucas Cheadle, assistant professor at Cold Spring Harbor Laboratory, Yvette Fisher, postdoc at Harvard Medical School, soon to be assistant professor at UC Berkeley, Fenna Creenan, postdoc at Harvard Medical School, Bianca Jones-Marlin, postdoc and soon-to-be assistant professor at Columbia University, and Cindy Poo, postdoc at Champlemal Research. Unfortunately, Cindy's not able to participate in Showcase this year, but we look forward to hosting her for a future event. Our program this year looks a bit different, as we won't be hosting our usual bustling poster session in the atrium. In addition to the Next Gen Leader Talks, we've asked teams across the Allen Institute for Brain Science and MindScope program to create reimagined project talks, which cover everything from the latest analysis of our data to pipeline modifications made this year to our data and technology teams' work to bring our vast data sets to the public on brainmap.org. As mentioned in a press release in April, neuroscience research at the Allen Institute is continuing to evolve. The Allen Institute for Brain Science underwent a transformation this year, with Honghui Zheng taking the helm as we prepare to embark on a new multi-year plan to build on our fundamental knowledge of cell types, the building blocks of the brain. Christoph Koch continues to lead our MindScope program, now a separate entity within the Allen Institute, which will continue to pursue understanding of the visual system. Today, we'll hear from our electron microscopy. Next slide. Today, on the brain science side, we'll hear from our electron microscopy and human cell types teams in the Allen Institute for Brain Science and the visual behavior team from the MindScope group. Next slide. Tomorrow, our project talks will feature the MindScope Neural Pixels team and our transcriptomics and transgenics team at the Allen Institute for Brain Science. And in the next slide, our final day of Showcase will have 4 project talks with synaptic physiology and patch-seq teams covering some of our latest releases from the Allen Institute for Brain Science, and MindScope's visual coding 2-photon team sharing their latest analysis. We will also hear from the data and technology team about their work supporting data releases and knowledge integration across both divisions. Before we get started, I think next slide. Before we get started, I'll just reiterate what Caitlin mentioned earlier. We will take live questions via the Q&A panel for our Next Gen leaders throughout Showcase. For all of the pre-recorded project talks, we won't have live Q&A, but we encourage you to submit your questions to the teams via our Allen BrainMap community forum. We'll drop this link in the chat regularly throughout the event, and you can find all the related threads to the Showcase 2020 tag. Finally, in the next slide, I think it's the next slide that we need. Finally, we'd like to especially thank the late Allen Institute founder, Paul G. Allen, without whom this institute, this event, and much of our research wouldn't be possible. Okay, without further ado, I'd like to introduce our first project talk. Every year, the EM group pretty much steals the show with their data sets describing the rich structure and dense connectivity details of the brain. This year, I'm sure, will be no exception. So I'd like to introduce the EM group.
Welcome, everyone, to this showcase and to this presentation on electron microscopy. My name is Nuno Massarico da Costa, and I want to start by thanking the showcase organizers for putting together this meeting, by thanking Clay Reed and all our team for the beautiful data that we're going to show to you today. There's just 8 of us here today, but there are many more that contribute to generating the data, the tools, and analyzing the data. And also want to thank particularly to our collaborators in the ARPA project, in particular the team of Sebastian Sung in Princeton, that generating this beautiful reconstruction segmentations of our data that you're going to see, as well as Andreas Tulias in Baylor College of Medicine. So cell types are one of the most fundamental concepts in neuroscience, and the electron microscopy can contribute to it in several ways, from the fine morphology of the organelles of a cell to its connectivity, but also is the morphology of single neurons. There are many other properties that our data sets cannot contribute to, as for example, transcriptomics or electrophysiology or synapse physiology of those same synapses that we see the structure. But since, and you hear a lot about these different properties in this showcase as well. However, and as you'll see, increasingly, a single cell morphology can also be collected in these other modalities, with these other modalities. And so single cell morphology can serve as a Rosetta Stone, as a link between the connectivity and organelle morphology that we're going to show you today, and all these other properties of neurons. So we chose electron microscopy because we were interested in mapping connectivity, and electron microscopy is still the gold standard to identify synapses at the structural level. However, to look at it at the scale of circuits, we had to also scale electron microscopy, and that's going to be the topic of the presentation of Wenjing Yin. Immediately after me, she's going to tell you how we put together this pipeline of several microscopes that can now collect EM data sets of the scale of circuits, millimeter cube size data sets with tens of thousands of neurons and hundreds of millions of synapses. And a particular data set that you're going to hear a lot is this one collected under the Microns program, which sits at over 4 different cortical areas in the visual system, which allows us not only to look at the recurrent connections within each one of these areas, as for example here in V1, but also the interareal connections, all at the synapse resolution. And in the next slide, I'll show you one reconstruction of one of the neurons in this data set. It's a layer 5 seam-tufted pyramidal cell, also known as IT for intercortical. In blue, that's the dendrite, and in purple, it's the axon. And as you can see, you can get this beautiful gross morphology of the neurons with millimeters of axons that can then be matched with reconstructions in other pipelines, but also you can zoom in to its synapses and then see who is the postsynaptic target. And here's one example of the thousands of synapses that this neuron forms. In this case, it's a layer 2-3 pyramidal cell in red. So as I mentioned earlier, we collected these data sets because of connectivity and morphology of neurons, but they are much deeper and richer than that. So we're going to start after Wenjing with some cellular biology. And Russell Torres is going to tell us about how we can use this data set to explore not so well-known organelle to the masses, the cilia, but every neuron has one, and it's fundamental, and it's been implicated in many neuronal diseases. So, so in blue here is the 3D reconstruction of some of the neuron, of the membrane of a neuron. If we peel it off and look inside, there's many more organelles. And Nicholas Stern is going to tell us about a particular one, the mitochondria, which again is crucial for the functioning of neurons, and also its synapse function. And synapses sit in many structures on a neuron. And Sharmi Shashamani is going to tell us about how we can use machine learning to identify the structures where synapses are made, like spines, shafts, and somas. And then this will be very useful to then later on help with the classification of these neurons into cell types. After this, we're gonna get into the connectivity between different cell types. First by Agnes Bodor, looking at the connectivity of layer 5 tufted cells, also known as ET or PT, both locally within V1, but also across areas. After Claire Gamlin is going to tell us about the connectivity of inhibitory cells, focusing on a particular subclass, the Martinati cells, which are also the ones that are positive for somatostatin. And then Joanne Buchanan is going to tell us about glial cells, because of course they are present in the data set, and the segment, and we also can extract beautiful segmentations of them. And she's going to tell us about exciting new finding that we got from the EM that provides a new view and a new role on oligodendrocyte precursor cells. Finally, we're going to zoom back into a cellular compartment, the soma with its nucleus, which you see here in these figures. And Forrest Coleman is going to show you how, even based on these very local features of a local part of the neuron, we can tie up a lot of what we told you before and characterize and classify cell types that is consistent with all the morphology that you've seen before. Finally, I want to say that one of our data sets is already public. Please use it and abuse it. We certainly have been having a lot of fun and excitement with it, and I hope you do too. And now I'm going to pass it to Wenjing. Thank you so much.
Hello, my name is Wenjing Yin. In the next few minutes, I will give you a quick overview of the EM imaging pipeline that we use to generate the data sets. After the histology and the sectioning team received the mouse, they produced a block and sliced it into 40 nanometer thin sections. The sections are then placed onto the specimen carrier called the grid tape. The tape is then loaded onto a reel and into the microscope. On the right side, you can see a photograph of the reel system inside EM suite. Inside the column of the microscope, there is a fast stage that can drive the specimen along the 2D scanning area and generate the montages. Working with Derek and Jay and also our external collaborators, we have added a lot of the modifications to the hardware and also developed our in-house software packages to enable this automated 24/7 continuous imaging. And currently, there are 6 systems of this kind in our lab. Over the past few years, we have generated several petascale EM data sets, which include IAPA Microns project. It's a 1 cubic millimeter volume of the mouse visual cortex covering the border of V1, RL, AL, and LM. And it took about 6 months of imaging time, which was done by the end of 2018. A year later, we did a whole bunch of upgrades and improvements to the systems and completed another 1 cubic millimeter volume centering the primary visual cortex of the mouse. And this year, over the summertime, we did a third data set, which is a human tissue on the medial temporal gyrus. And on the right side, you can see a 2D montage spanning 2.5 millimeter long, and the block is about 400 microns in thickness. And here is a live recording of parallel imaging across 5 microscopes. And because the user interface is web-based, we can actually access the real-time imaging remotely and also be able to control the microscopes. With that, I will stop for now and pass it to our teammates to discuss the interesting science out of those data sets. Thank you. If you have more questions, feel free to ask at the Q&A session.
Thank you, Wenjing. I'm Russell Torres, and in a project done in collaboration with Carolyn Ott at Janelia Research Campus, we've leveraged the contrast and resolution that make EM a classic method for studying organelles, along with the unprecedented context provided by our modern connectomics datasets to study primary cilia in the cortex. These organelles, which you can see in orange, are cellular sensory antennae, and in the mouse brain have been implicated in pathways for eating, learning, and behavior. We started by characterizing the ciliary properties of different cell classes in an exhaustive morphological study of our P36 layer 2-3 IARPA microns dataset. And this is by no means a small dataset, but since there's usually at most 1 cilia per cell, we were interested in adding from a growing library of available EM volumes for scale and to help build understanding about across cortical depth and age. Simply enough, we find first that the microglia and oligodendrocytes lack ciliary structures altogether. However, neurons overwhelmingly have a single cilium located at the surface of the soma. Among neurons, pyramidal cells have a distinct directional bias in the pia direction, which is not shared by the collections of interneurons or glial cells. Interneuron cilia also tend to be longer than pyramidal cilia, even at similar soma sizes. Astrocytes are often ciliated, but there are distinctions among these cilia in which some are anchored at the soma, like at the soma surface like neurons, while some have what we call a pocket region of the membrane, effectively reducing the surface area exposed to the neuropil. And the last classification, the oligodendrocyte precursor cell, which you'll hear more about later from Joanne, is an extreme example of this, where the exposed portion is usually exceptionally small. At most, we've seen it be about a micron. Now, beyond characterizing the morphological properties of cilia, the EM datasets give us a framework to conduct spatial analysis on the neuropil and allow us to ask some hypothesis-generating questions regarding the origin of ciliary signaling in the cortex. From left to right, our analysis includes ratios of contact between the cilium and adjacent cells, distribution of synapses around the cilium, and the distribution of nearby dense core vesicles. Next, Nick is going to describe the effort to use our datasets to analyze another important organelle, the mitochondria.
Hi, I'm Nick from Princeton, and I'll be talking about how we've used the IARPA data to study neuronal mitochondria. Neuronal mitochondria are not only responsible for generating the vast amounts of ATP required by synaptic transmission throughout the brain, but they're also intimately involved with calcium signaling within neurons as well. Understanding their distribution, morphology, and function might in turn give us a better understanding of neuronal function. Let's start by taking a quick look at an example EM image from our dataset. It's fairly easy to resolve mitochondria as they're very dark objects. In order to capture their morphology accurately, our group trained a machine learning classifier to find the image voxels that depict the mitochondria using supervised learning. We applied this classifier to the entirety of a P36 mouse dataset and segmented the predictive voxels into discrete organelle predictions using similar techniques to our synapse detection and nucleus detection. This yields a highly detailed picture of mitochondrial morphology throughout each cell in our volume, as depicted here. One strength of this method is that we were able to resolve dense structures within the somatic cytosol, unlike fluorescence techniques. In addition, we can leverage the other computational tools we built in this project to perform detailed analyses at scale. As one example, here I'm showing the computational skeleton of a cell, which simplifies neuronal morphology down to a linear structure. We can associate each mitochondrion with the skeleton to label it with a precise location and extent throughout each neurite. If we also label the skeleton by features like neuronal compartment, we can then propagate those labels to the mitochondria easily. Here, the green locations specify the axon of the cell, the orange show the basal dendrites, the red locations show the branches of the apical dendrite, and the blue specifies the soma. One thing that's known in the literature of neuronal mitochondria is that dendritic mitochondria are long and filamentous, while axonal mitochondria are short and punctate in pyramidal cells. You can readily see this in our data as well, as shown here within this inset. The dendritic mitochondria are teal here, and the axonal mitochondria are red. In addition, we recently found that the somatic mitochondria are statistically intermediate in size between these two populations. Neogenesis of mitochondria occurs in the soma, so this result suggests the existence of processes that transform the somatic mitochondria into the shapes we see in the neurites. Mitochondria are also suspected to be very important for postsynaptic transmission. Postsynaptic transmission is very energy costly, and calcium signaling at these sites might be important for information transmission, learning, or both. We see a signature of the connection between mitochondrial synapses within the dendrites of the layer 2-3 pyramidal cells of this data set. Specifically, the number of synapses the cell receives on its basal arbor is highly correlated with the density of mitochondria within that arbor. This is an interesting result, but it's somewhat coarse. Looking across the entire arbor, these synapses lump together hundreds or thousands of diverse connections. We see some hint of this diversity by looking at the dendritic spine morphology. Up next, Sharma will show you some of how we're trying to understand this exact diversity. For more details on this analysis, as well as several others, please refer to our preprint on bioRxiv. Thanks for your time.
Hi, I'm Sharmishta Seshamani, a scientist with the EM Connectomics team. And here I will talk about postsynaptic regions and how they can be used to describe cells. As Nick mentioned, there are lots of different types of synapses. What we're interested in here is the local region on the postsynaptic cells around a synapse. We call these regions postsynaptic shapes, and they can appear as spines, dendritic shafts, or local regions on the soma. What this analysis aims to do is to generate a description of each cell based on its postsynaptic shapes. To start, we extract all the postsynaptic shapes for a set of cells and create a dictionary of shapes by training an autoencoder to learn a feature representation for each shape. When a set of these is collected, we can use dimensional reduction techniques such as UMAP to look at the space that they cover. The video here shows what we can get from such a dictionary. You can easily observe that spines carve out their own region in the top part of the space. And so do shafts in the middle region, and soma in the lower left. Furthermore, note that the space is continuous, and the variability of shapes within each of these types can also be seen as you move along the space. This dictionary can now enable us to describe a cell using its postsynaptic shapes. Here are examples of postsynaptic shapes present on two types of cells. On the left, we have a layer 5 pyramidal cell, and on the right, we have an inhibitory cell. It's very easy to see the differences in appearances of the cell reflected in the maps below. We then quantize this space to generate a numerical representation for each cell and perform a data-driven clustering of 1,835 cells. Here is a result of such a clustering. What is immediately observable is that we're able to separate most inhibitory cells from excitatory cells. We can further analyze the different groups here and also combine these postsynaptic descriptors with other unique features obtainable from EM data to investigate neuronal subtypes. To learn more, please come to our Q&A session. Thank you very much. I will now pass it on to Agnes, who will discuss her work in analyzing connectivity between cells.
Hi, my name is Agnes Bodor, and I am a scientist at Allen Institute. In this study, I will introduce you to the local and long-range connectivity of layer 5 thick-tufted cells. Here I show you our dataset on a top-down view, and as you can see, it's enough big to study long-range connectivity between primary and higher-order visual areas. In this video, I show you a randomly selected pyramidal cell colored dark blue and its long-range axon crossing through our dataset, and with other colors you can see many of its target cells reconstructed in 3D. In our study, we choose layer 5 thick-tufted cells, or on other names, ET or PT cells. This cell has a large soma, thick apical dendrite, and a large tuft in layer 1. It's also famous because it has a connectivity outside of the cortex, but it also has local and long-range axon within the cortex. Here I show you that we proofread partially the axon arbor, both locally and long-range. And then we found all the synapses on this axon arbor, and we labeled the synapses based on the target element. If the target element was inhibitory cell, we used red, and if it was excitatory cell, we used blue dots. Here I show you only the local arbor and the synapses labeled with red and blue, and you can see about 2/3 of the synapses were on inhibitory targets, while only 1/3 was on excitatory target cells. If you look the same on the long-range axon, you can see the opposite. About 2/3 of the target cells were excitatory and only 1/3 was inhibitory. Of course, this dataset is awesome because you can look every single target cell, and based on their morphology and location, you can define the cell type. Based on the statistic, we found that most of the target cell, when it's excitatory, is layer 6 cell, but sometimes layer 5. And when the target cells are inhibitory, it's usually basket or Martinati cells. This is true both locally and long range. In the next talk, Claire will talk about inhibitory cells and their connectivity especially about Martinati cell.
Thank you for that introduction, Agnes. My name is Claire Gamlin, and I am a scientist here at the Allen Institute. Today, I'll be telling you about target specificity of T-shaped Martinati cells. Martinati cells are somatostatin, or SST, interneurons, and a recent paper from the Allen Institute found that in mouse primary visual cortex, there are at least 13 distinct SST MET types. And here, a MET type is defined as having congruent morphology, electrophysiology, and transcriptomics. Of these 13 types, at least 2, type 6 and 7, can be considered T-shaped Martinati cells because they send axon up that then branches in layer 1. These 2 T-shaped Martinatis, however, differ in their morphology. Type 6 has a soma deeper in layer 5 as compared to type 7. Type 6 branches while in layer 2-3, whereas type 7 branches close to the layer 1-2-3 border. And within layer 1, type 6 has sparse axon that reaches up to the top of the layer, whereas type 7 has very dense arborization in the lower half of layer 1. So my goal was to look in the EM dataset to see if I could identify these 2 distinct morphologies and look at their postsynaptic targets to see if there were any differences. Here I'm showing what I think are 3 type 6 Martinati cells. They have somas deeper in layer 5, they branch while in layer 2-3, and send axon up to the top of layer 1 that is sparser compared to type 7. I can take an example one of these cells and identify synapses that it makes in layer 1, as indicated here by these green dots, and then trace these connections to the locations of the postsynaptic targets. And here in cyan are the soma locations of the excitatory postsynaptic targets of those synapses. So for this cell, it makes synapses onto layer 2-3 pyramidal cells, 2 layer 4 pyramidal cells, and several layer 5 pyramidal cells. And for this cell, the layer 5 pyramidal cells are all PT cells. I was also able to find examples of what I think are the type 7 SST MET type. They have somas higher in layer 5, they branch close to the layer 1-2-3 border, and within layer 1, they have very dense arborization. Again, I can identify synapses that this cell makes within layer 1, as indicated by these green dots, and look at the location of the postsynaptic targets. Here, we can see that this cell also makes synapses onto layer 2-3 pyramidal cells, 1 layer 4 pyramidal cell, and several layer 5 pyramidal cells. And all of these layer 5 pyramidal cells are actually IT cells. So not only are we able to find these 2 distinct morphologies, but our preliminary data suggests that their targeting is different with respect to the layer 5 pyramidal cells. There aren't just neurons in this data set, however, there are also glia, and Joanne is going to tell you a little bit about the exciting results she's finding in the EM.
Hello, my name is Joanne Buchanan, and today I will be telling you about a new role for oligodendrocyte progenitor cells, also known as OPCs. In our data set, we found 4 types of glia: oligodendrocytes, OPCs, astrocytes, and microglia. Most glia, except astrocytes, can be found in a satellite position on neurons, that is, with their somas closely opposed to the host cell. In this case, in gray, we have a pyramidal cell, a microglia in purple, and an OPC in pink. OPCs can differentiate to become myelinating oligodendrocytes or remain in an undifferentiated state throughout their lives. What role they might play in the adult brain is not yet clear. When we took a closer look at the branches of the OPCs, we could see within them a large number of lysosomes and phagolysosomes. This was an unexpected finding. In addition, when we look inside the chambers of the phagolysosome, we're able to identify some of the components, in this case, 40 nanometer vesicles. In order to corroborate our findings, we collaborated with the Dwight Burgos lab at Johns Hopkins. They used lamp staining to stain the lysosomes and phagolysosomes within the fuchsia OPC in a cultured cell. In addition, we counted the number of phagolysosomes in both the microglia and the OPCs in our 2 data sets. we were surprised to find that the OPCs had significantly greater numbers of phagolysosomes than the microglia, who are considered to be the professional phagocytes of the brain. We then took a closer look to see what was ingested within the OPC branches. Here shown an axon in green encapsulated within the cytoplasm of the OPC in pink and 2 phagolysosomes nearby. The 3D rendering shows the collateral branch of the axon here in white arrows, and again with the OPC removed shows how it was completely ingested inside the OPC. We were also able to identify the types of axons that were ingested because they were segmented. Here we counted the numbers and types of branches and pieces of axons ingested in 2 different cells, and we were able to find out that more than 80% of the ingested axons were excitatory. This suggests a new role for OPCs in circuit refinement by pruning excitatory axons. With that, I'll pass the baton to Forrest, who will show you about how nuclei can be used to identify cell type.
Hi, my name is Forrest Coleman, and I'm an assistant investigator here at the Institute. Over the last 30 minutes, you've heard a variety of talks about how the EM data can be used to provide insight into the structure of cells, from subcellular organelles to their long-range connections across brain areas. And this figure here illustrates that point yet again. In the middle, you can see the dendritic reconstruction of a layer 5 excitatory cell. And then on the right, you can see a zoom in of its somatic region. And on the left, you can see a zoom in of its apical dendrite in layer 4. And as I hit play on this movie, you can see the cells rotated around, and every few frames, you'll see a new cell. What's striking about this video is how diverse these layer 5 excitatory cells are, and how that diversity is expressed both at the level of their difference in dendritic morphology and branching patterns, as well as the shape of their somatic regions, and the caliper and character of the spininess of their apical dendrite in layer 4. Here at the Institute, we're interested in combining all of these information across all these different length scales to make quantitative measurements about cells, ultimately to describe how are different cell types connected to each other in cortex. But as we start along that journey, where do we begin? Well, we begin at the nucleus. We've developed a pipeline for segmenting the nucleus separate from the cell membrane that lets us quantify various aspects of the somatic region of cells. On the right is 2 such aspects, namely the area of the cellular membrane in and around the cell versus its nucleus volume. And you can see in this space that the major cell classes separate quite nicely. The astrocytes are over here. The OPCs you heard about from Joanne are here. The neurons are all in a diverse cluster here. You can see oligodendrocytes down here and the microglia down here. So we can look in more detail, though, at the nucleus. Sometimes nuclei have interesting structures, such as these folds that are shown here in orange in 3D across a variety of different cell types, or in 2D here in a 2D EM picture, with, again, the folds highlighted in yellow. And this lets us measure things like what fraction of the membrane area are within one of these folds as a function of depth of that cell within cortex. You can see the blue dots, which are excitatory cells, have this characteristic pattern in which they get more and more folded as you go deeper, and then they're less folded in layer 6. This is a contrast to the inhibitory cells in purple, in which are relatively equally folded across all layers of cortex. So these are just 2 features that we can measure about the cell. If you want to hear about more features that we can measure, please come to our Q&A session. But I'm just going to leave you as a teaser with this UMAP plot. It's a low-dimensional projection of many, many features measured across 70,000 cells in one of our larger data sets. And now you can see that they're colored with respect to the output of a machine learning algorithm that's attempting to classify what type of cell they are. So with that, I'd like to thank everyone at the Allen Institute who helped make all these data possible and all the talks that you've heard over the last 30 minutes. As well as our colleagues in the Sung Lab from Princeton, who are largely responsible for the beautiful segmentations that you saw, as well as our colleagues at Baylor, who help us make functional measurements of these same cells. And of course, our founder, Paul Allen, who we thank for his vision, encouragement, and support. Thanks.
Thanks, EM group. That was amazing as usual. As a reminder, we won't have live Q&A for the project talk, so please attend the Q&A sessions that you would like to ask questions in, and please visit the forum to submit a question for this team. We have dropped the link in the chat for your convenience. It is now my pleasure to introduce our first Next Gen Leader talk. I'd like to introduce Bianca Jones-Marlin. She is an incoming assistant professor of psychology and neuroscience at Columbia University's Zuckerman Institute for 2021. She is a neuroscientist and postdoctoral researcher at Columbia University, presently in the laboratory of Richard Axel, where she investigates transgenerational epigenetic inheritance or how traumatic experiences in parents affect the brain structure of their offspring. She holds a PhD in neuroscience from New York University and dual bachelor degrees from St. John's University in biology and adolescent education. As a graduate student, her research focused on the vital bond between parent and child, and she studied the use of neurochemicals such as the love drug, oxytocin, as a treatment to strengthen fragile and broken parent-child relationships. Bianca aims to utilize neurobiology and the science of learning to better inform both the scientific and educational community on how positive experiences dictate brain health and social well-being. Her presentation is titled, Bridging the Gap Between Innate and Learned Behaviors, a Parent's Role in Promoting Survival, and will discuss how transgenerational epigenetic inheritance of environmental information remains a complex mystery. Welcome, Bianca.
Thank you very much for the introduction, and thank you for coming today. I'm excited to speak with you about my work on neuromodulators and the relationship between parent and offspring, and looking at how we can bridge the gap between innate and learned behaviors. My work focuses on the relationship between parents and their offspring, and that essential bond being necessary for the survival of offspring. And as parents can perpetuate this survival in one of two ways. We perpetuate this innately by passing on instinctual and genetic information to our offsprings that allot for survival, as well as learned instruction and support via experience that allows our offspring to navigate the world. An example is a child. Anyone who's hung out with a child before knows that they do enjoy putting things into their mouth, although the things that they put into their mouth may not always be something that's beneficial for their survival. We as parents and educators aid children to look for things that are going to be beneficial for them and which they can eat. And here's an example here of a primate showing their offspring how to forage for good food that's beneficial for them and their survival. I look at this relationship in two ways. I explore if an innate behavior, like the innate behavior to put something into your mouth, can be learned and optimized. And I look at this through learning of maternal behaviors and how that affects the survival of the offspring. I also then look at the other part of this question. Can a learned behavior of a parent then become innate in an offspring? And I look at this focusing on paternal adaptation to threat and how this can be inherited in the offspring for survival. First, focusing on maternal behavior. If a child begins to cry, it's usually that a parent, in this case, a mother, will go towards the crying baby, pick them up, and take care of them. Similarly, mice also perform this task. When pups are removed from their nest, mice pups, they begin to cool rapidly because their inability to thermoregulate. And they emit something called an ultrasonic vocalization, which is above our hearing range. And this alerts the mother. So the mother will orient towards the pup. She'll pick the pup up and she'll bring the pup back to the nest. And I'll show you a video of that here. The pup is removed. The mother will orient towards the pup, although she leaves the rest of the pups in the nest, and she'll bring it back. That's a successful retrieval. Virgins, however, usually won't perform this task. They'll ignore pups, and at times they'll cannibalize pups. We see a pup is removed from the nest, and this is a virgin mouse. And although the pup is emitting ultrasonic vocalization, the virgin does not retrieve. And herein lies my question. How does an innate existing cue, in this case, the sound of a pup crying, elicit a response in a mother who has had the experience of birth, but not in a virgin? And can this call unlock an innate response in a virgin through learning and elicit the essential behavior of retrieval for the survival of the offspring? And I focused on the neuromodulator oxytocin in answering this question. And I focused on oxytocin because oxytocin is known and essential for sex, for pregnancy, to maintain pregnancy, for the birthing process. It induces uterine contractions. It's essential for breastfeeding, milk letdown. It's released in humans and mammals with soft touch and eye contact. It has been shown in rodents to increase maternal behaviors such as burrowing, building a nest around pups, and also orient towards pups. I asked 3 questions in relation to this question of innate and learned of maternal behavior. I asked what? Can oxytocin, given its important essential role in the birthing process, induce retrieval in virgins? I asked, where is this taking place? Considering the sound of a baby crying orients the mother towards a pup, I then asked, where? Is the auditory cortex the locus of action? I then asked, how? If this is the case, how does oxytocin induce changing in A1 or primary auditory cortical areas after learning retrieval? And so to focus first on what can oxytocin do, I performed a paradigm where I have a mother and a virgin and in a behavioral arena with the mother's pups. I then remove the mother and I take a pup from the nest and I give the virgin an opportunity to retrieve the pup. Once she doesn't retrieve, I treat her with oxytocin. She'll either get an intraperitoneal injection of oxytocin, a saline injection as a control, or I'll stimulate the paraventricular nucleus of the hypothalamus. This is an area of the brain in which oxytocinergic neurons reside, and I can release endogenous oxytocin into the brain's milieu. She's tested every 3 hours for 3 days and given 10 minutes to retrieve a pup. Here's an example of a virgin who has been treated with oxytocin via the optogenetic stimulation and the endogenous release of oxytocin after 12 hours of being co-housed with a mother and her offspring. And we'll see as I stimulate endogenous release of oxytocin, we see that this virgin has become a successful retriever and will now bring pups back to the nest. Here's an example of our graphical data in which animals that were treated with oxytocin in red and optogenetic stimulation of oxytocinergic neurons in blue retrieve at a higher rate. A larger percentage of them are retrieving than compared to saline. And this is through the systemic treatment. Therefore, endogenous oxytocin can enable retrieval. Oxytocin induces learning of retrieval behavior in virgins. We then want to ask, where is the auditory cortex the locus of this action? In order to do that, I, along with my colleague Mariella Mitri, created a novel antibody to the oxytocin receptor. And what we discovered was that oxytocinergic receptors were found represented in the mouse brain. That's shown here graphically by any area that is red having higher percentage of oxytocin receptors. And what we found was astounding when we stained for oxytocin receptors in the primary auditory cortical areas. We showed that in the left auditory cortex, we saw a higher representation of the oxytocin receptor compared to right. And that's shown here graphically, where on the left of both virgins and mothers, we saw more oxytocinergic receptor presenting neurons than on the right. And why this was so exciting is because there was a lateralization to the left auditory cortex. And this lateralization potentially could mirror what we observe in humans, where in humans, most right-handed people have a Broca's and Wernicke area, areas of expression and understanding language, located on the left side. So to have a social area associated and lateralized in the mouse was extremely exciting to find. We wanted to know if left auditory cortex was then essential for pup retrieval. In order to do that, I use the GABA agonist muscimol, which means I'm just transiently inactivating left auditory cortex. And I'll show you a video now of a mother who was a former excellent retriever, then being treated with muscimol. She has a cannula in her left auditory cortex, which is a small straw in which we can deliver the drug. The pup is removed from the nest, and the mother hears the vocalization and retrieves the pup. This is after the treatment of muscimol. You'll observe that the mother seems to observe that the pup is outside of the nest. And yet it's as if that cue is not processed due to the silencing of the left auditory cortex. Given the importance of the left auditory cortex in this behavior, we wanted to ask if oxytocin itself in left primary auditory cortical areas could induce retrieval behavior. In order to do that, I used the cannula and delivered oxytocin directly to the left auditory cortex, or saline as a control, or I optogenetically activated the terminals of the oxytocinergic neurons that project from the paraventricular nucleus to the left auditory cortical areas, shown here in green by their green terminals. And what we observed was exciting. We saw that oxytocin just in left auditory cortical areas could induce this retrieval behavior, similarly to what we observed when it came to the systemic treatment. So oxytocin in left auditory cortex can enable retrieval behavior. The left auditory cortex mediates oxytocin-induced retrieval. Therefore, how? How does oxytocin induce changes in primary auditory cortex after learning of retrieval? In order to do this, I used the technique, in order to answer the question, how does learning change cortical responses to pup calls? I used a technique called whole cell in vivo physiology, in which I'm recording from single neurons in the primary auditory cortical areas while playing the pup calls. I recorded a library of pup calls as shown here in a spectrogram, plotted a frequency, usually on the right, on the y-axis over time. And we played the sound of a pup crying. Although we didn't have an actual pup there, we wanted to control for smell. We actually presented these recordings to the mother who was anesthetized. Playing the pup calls, I then patched single auditory cortical neurons and looked for, in these examples, a spiking output via current clamp. Here's an example of a mother's response to a pup's call. What we see here is a beautiful time-locked response to a pup call. On the raster plot on the bottom, every spike that is in blue, every dot that's in blue, represents a spike that fell within 10 milliseconds of the previous spike. And why I highlighted this is to show the robustness and the reliability of this response in a mother of the single cell in the primary auditory cortex. Here's an example of a call from a pup and a response to naive virgin. You'll note that this call is different than the prior call. I chose the best response to pup call for every cell that I recorded from. And we see that this neuron responds. We don't see this robust and reliable response that we observed formerly with mother. However, after a virgin learns to retrieve and exhibits this successful retrieval behavior, we see these changes in the primary auditory cortex, where a former scrambled response that we observed in another naive virgin seems to align to the representation that we observe in a mother, a time-locked and robust response. How could this possibly be happening? In order to answer this question, we performed a whole cell in vivo physiology patch and looked for the synaptic input, so the excitatory and inhibitory inputs into these cells via voltage clamp recordings. And here's an example of a pup call being played. And here is my my excitatory input recordings. And what's exciting that we observed is when we look at the inhibition and excitation, it's balanced in the mother, as in the inhibition and excitation are both emerging at the same time of the call. But I'd like to point you to this little node right here, which we see that excitation precedes inhibition just a little bit. And this could be leading to our successful spikes that we see in mothers. In naive virgins, we see excitation and inhibition, but it does not seem to be balanced or time locked. However, after a virgin expresses retrieval behavior, we see this reemergence and rebalancing of excitation and inhibition, always with excitation preceding inhibition, potentially leading to those time locked spikes. So how does a virgin brain turn into a mother's brain in real time? In order to answer that question, I did whole cell in vivo physiology, and I essentially mimicked the experience of childhood, of birthing, potentially in the auditory cortical areas. In order to do this, I played the sound of a pup call, recording from a virgin. This is a virgin mouse I'm recording from who is not expressing retrieval behavior. After 15 minutes of recording this one cell, I then optogenetically activated the terminals in the primary auditory cortex and paired that with the sound of a pup crying. Over 30 minutes of recording from this cell, we start to see an increase in spiking. And after 3 hours of recording in this area and patching onto another cell in this area, we see the emergence of this time-locked response. We essentially took the experience of motherhood by flooding the primary auditory cortical area with oxytocin, and then pairing it with the first salient cue a mother could experience after birth, the sound of her baby crying. And it led to this time-locked response that is a signature of the maternal response that we observed. So I showed you how synchrony via rebalancing of cortical excitation and inhibition led to these responses. And so we took an innate behavior, the behavior of pup retrieval and a mother for their pups for survival, and showed that this could be learned in a virgin. I then wanted to observe the other part of this bridge. And that's looking at how learned behaviors in a parent can become innate in the offspring. And this work stems from studies done in humans during the Dutch hunger winter. And during the Dutch hunger winter, this is the last winter of World War II, the Netherlands were cut off from food and relied on food drop-offs, as shown here in this picture, from other countries, also including the United States, in order to allow for their population to survive. And given that the Netherlands took such good note of those that served in the military, a trend started to emerge. Those who were offspring whose parents had suffered during the Dutch hunger winter, specifically males whose parents had suffered, males who had suffered during the Dutch hunger winter, their children and their grandchildren suffered from metabolic issues, hypertension, which is high blood pressure, diabetes, and even schizophrenia was noted. Other labs have looked at how this could be replicated in mice and what actually could happen in which a memory of starvation in a parent could be passed down to an offspring. Oliver Randall's work at University of Massachusetts has done studies on low protein, high fat diets in mice and observed their offspring also have altered metabolism, reproductive success, etc. Work done by Carrie Ressler's lab at Harvard has shown that if you treat a mouse with an odor and shock that mouse with an odor, they elicit changes in the F1 population, the Cilia 1 population, where those children will avoid the odor. And they've also shown changes in the olfactory areas. I wanted to follow up on this work. In order to do that, what we did is we performed the odor paradigm in which we presented an odor, acetophenone in this case, Laurel, which smells like flour, and propanol smells like alcohol. We did a paired and unpaired experiment where we present the odor for 10 seconds and either co-terminate it with shock for paired, or we have an unpaired where we present the odor for 10 seconds, wait a minute in between, and then shock. So all of the animals are getting shock and odor. You'll note in this video that on top, the acetophenone paired animal then avoids the acetophenone side, that's the red box, whereas the acetophenone unpaired has had acetophenone and shock, but there's a minute in between. And that mouse does not avoid the acetophenone chamber. This is a successful and reliable treatment of, for odor avoidance. We then sought to look at the main olfactory epithelium of the mice. And that's shown here, where we have cells in the main olfactory epithelium. And these cells project to all cells that express a receptor. There's a 1 neuron, 1 receptor rule. And these cells then project to their one glomerulus. I performed eye disco, which is brain clearing, in order to observe these cells. As I showed you in the previous picture, we can see beautiful cells and we can see these glomeruli, but we can't see changes on a single cell layer. So in order to really dive into that, we use brain clearing. Here's an example of tissue. Prior to clearing, I then embed it in agarose, and I can clear the brain while maintaining the fluorescence and being able to use immunolabeling. I can look at single cells in the entire tissue. And here's an example here in which I have cells that respond to Lyral. These cells are the MOR23 GFP main olfactory epithelium neurons that are in purple. And green, we have M71, which is another receptor that we found in these neurons. And using these techniques, I'm able to look in the parent at what is actually happening on a single cell layer, on a single cell level. Are we seeing an increase in cell number which work we're following up on. And how could that be happening? In which a odor paired with a shock is actually changing the epithelium of the nose. I then observed, using something called EDU, which is a birth dating method, how cells could be living longer. Are more cells being born that express these receptors? Or is there something called receptor switching, when this one neuron, one receptor rule is then broken because of a traumatic experience, in this case, the foot shock? And so here's an example here of 2 images that we took in the main olfactory bulb. To the left, we have EDU treated for 3 days, and on the right, EDU treated at 21 days. And these cells labeled in red are the rostral migratory stream, and we can see over the 3-day and 21-day point where these cells, when these cells are born and where they move to, allowing us to track new neurons that are being born, where they're going, and to note whether or not cells are living longer or more cells are being born in our main olfactory epithelium in response to this shock. Moreover, we're able to now look not just at the paired and unpaired of the fathers. I'm specifically looking at fathers in this case. And here's an example of a main olfactory epithelium in which I'm pointing out the green cells that respond to viral and the red cells that are birth date, but also in the offspring. There's a traumatic response, a pairing of a shock and an odor in the parent lead to changes in the epithelium in the parent and also in the offspring. And how could that happen? And that's work that we're following up now. And I'm excited to show you that data hopefully soon, as well as work that I'm following up on and going to start my lab in Columbia starting January 1st. And in my lab, we're going to focus on these questions, looking at experience-driven changes in the main olfactory epithelium. Given the shock and the odor, what are the lasting epigenetic changes in mature neurons and also in the stem cell population of the nose? I'm also going to look at how 1 minute can make all of the difference, given that the paired and unpaired have a very similar experience, but the 60 seconds in between could be leading to the difference in changes that we have been observed that I didn't go directly into today, that have been observed in the main olfactory epithelium, as well as changes that we observe in the F1 population. And how could neuromodulatory representations, for example, stress-induced neuromodulatory regulation, change the MOE? And thirdly, what are the mechanisms of inheritance? What is the epigenetic inheritance and adaptive traits, and how are they passed on? Here's a video that we took from a mouse that's anesthetized, and we're able to take sperm at different time points and look at the RNA responses in changes to the traumatic experience. And so with that, I'd like to thank those that worked in oxytocin, specifically Robert Frumke, my advisor, and those that are following up with the work now, as well as my amazing group of technicians and undergraduates who worked with me with my PhD, 2 of which are with my postdoc, 2 of which are coming with me into my own lab, who've done a lot of the work looking at transgenerational epigenetic inheritance of trauma. And with that, I'd like to thank you for your time and happy to take questions. Thank you.
Thanks, Bianca. Really fascinating. So if you want to ask Bianca a question, go ahead and submit in the Q&A. We already have a few, but I'm going to take this opportunity to take advantage of my moderator position and ask a question. I was kind of fascinated by the increase in proliferation or cellular production in the olfactory bulb, and I was wondering if you had any clues as to when that gets started and what the signal might be for more cells. So are they already dividing in the rostral migratory stream, or do these progenitors divide in the olfactory bulb, or is it in the SBZ?
Thank you for that question. So I showed you the rostral migratory stream to show how EDU can so beautifully trace and track a neuron. We're looking in the main olfactory epithelium. So these are where the cell bodies are going to lie, and they're going to send their projections to the bulb. And in the main olfactory epithelium, we have both a immature cell layer and then a mature cell layer. And so we're really looking at what could possibly be happening to lead to this increase in cell number. Could it be that the mature cells that are being active and also firing at the same time, so a coincidence detection with the neuromodulatory component, leading to some change in which they live longer, they're now going to be more available. So when new cells are born from the stem cell population, we see an increase in cell number. Or are these cells somehow sending a message, as in their dendrites actually go through the immature cell layer, in order to get to the nasal cavity? Are they sending a message to some cells that bias their receptor choice? This one receptor, one neuron rule, saying you are going to be this purple receptor, but please, we need you to become a green receptor for survival. Or thirdly, and this is we're excited to see where this goes. Could a cell that originally was expressing this purple receptor, the MOR23 receptor, then be switching their receptor choice? And this really just breaks the canon in which we have this one receptor, one rule. But immature cells, as they develop, they are expressing one of a few receptors before they choose their one receptor to express at the membrane. And so could it be that we can bias that receptor choice in a mature cell by having them cell switch?
Cool. Interesting. So, all right, we have a question from Michael Williams. Michael Williams says, hello, Bianca. Did you see that your oxytocin receptor 2 antibody picked up intense staining in CA2 as well?
Yes, we did see intense staining in CA2. So happy to talk to you more about that if that's your area of interest for sure. We did see staining in CA2. And this work is published, and this Mariella Mitri is the first author in this work, published in JNeuro. So she has a beautiful representation of oxytocin receptors in males, virgins, and mothers, and also at different age time points. She did a beautiful job with that.
Okay, we have a question from Julie. Her question is, was the 1 minute late shock delivered while the mice were still in the acetophenone zone? And does a pre-odor shock also have any effect? i.e., does the order of causality matter?
So the animals, thank you for that. The animals are in a chamber in which I have a fan blowing the odor through. So there's 10 seconds of the odor being presented. And one of the caveats of working with odor is that it's hard to really make sure that all odor is absent in the chamber. But the pre and post really becomes mixed in this paradigm because we're giving the odor 5 times and giving shocks 5 times within this training session. So at some point it's the odor and then the shock and then the odor and then the shock. So the before and after may become mixed to the mouse in between this time. But we are hoping that the concentration, at least the highest concentration point, is at the 10 seconds of the first odor presentation co-terminating with shock, which means the 60 seconds in between, we are seeing a lower presence of the odor. So the pre and post does not seem to be as important in the unpaired, and in the paired it is, because we only see that behavior in the paired where the odor does say that the shock is coming. It predicts the shock.
Okay, next we have a question that might take some time to answer from Ben Slivka. He says, fascinating, does this mean Lamarck was right?
This question is going to get me. I usually try to defend Lamarck because why? Lamarck came well before Darwin and only used what Lamarck had in this process and what Darwin built upon. For those who may not fully know, so Lamarckian inheritance, an example of this is the giraffe, in which the giraffe says, I really want to get to the top of that tree, so I'm going to try really hard and stretch my neck to get to the top of that tree. And I get the top of the tree, I get my leaves, and my offspring are born with longer necks. Whereas Darwinian inheritance says, I was born with a longer neck, I'm going to eat from the top of all the trees, and my short neck cousins will all unfortunately die, and I will perpetuate on. And therefore, I will pass this long neck on. And I think the beauty of looking at both of these, Lamarckian and Darwinian inheritance, is that both are represented right here in these data, in which an experience, which is still modulated by genetic cues, could be leading to the plasticity that leads to survival, and survival being the end goal. So I will plead the fifth on who's correct when it comes to that. But I do think both help edify where these data are going and what they're showing.
Cool. Okay, so Eric Yatri asks, was there a null control for imprinting using either a non-pup or non-distress sound?
Using a non-pup or non-distress sound. All of the sounds that we recorded were distress calls. So distress calls usually fall between 40 to 60 kilohertz, and they usually fall within, I would say 40 to 60 kilohertz. And so we specifically focused on these stress calls. And yes, the non-pup, the beauty of using an auditory cue is the reliability. We did not always use pups In other experiments we did, we used just the sound of the pup, and we saw the same orientation towards the speaker, in which the mom would go and search around the speaker for the pup. So we really wanted to control for the fact that it wasn't just the smell that's inducing this, or not just the visual cue that was inducing this. This retrieval was also done under red light, so we're not relying on visual cues. We specifically wanted to make sure that the auditory cues were what were at least leading to the orientation towards the pup.
Okay, next we have a question from Forrest Coleman. He says, "Hi, Bianca, fascinating talk. I'm curious if you could enumerate some of the proposed mechanisms for storing these inheritance effects, gene expression changes in gamete cells, or that it's transferred from the mother during embryogenesis, or is there some other possibilities?"
Thank you for that question. Specifically in the second part of the talk, the first part I really focused on mothers purposefully, and the second part I focused on fathers purposefully. And this is because although it is a beautiful and exciting study to look at how a pregnant mom, for example, could go through a trauma and look at the offspring and the grandchild of that. We can use this example to also see that if a mother is pregnant with her daughter, essentially the eggs of her granddaughter would be present if she were to have a daughter 3 generations down. And I really wanted to not focus on that. I wanted to focus on the fact that this memory could live on in something that may not necessarily need to be present when the embryo didn't need to be present, and that's sperm. And my sperm are going to turn over within 21 to 40 days. And yet at this 21-day time point, we're still, sorry, that was my timer. We're still seeing this response. And so the question goes back to what could be happening when it comes to the sperm. The specificity we really believe lies in RNA. To have a receptor choice be so specific, we really are focusing and using RNA sequencing to really parse out what could be happening in the male olfactory epithelium in these living longer / potentially switched cells / potentially being born cells, and also what message of that component is maintained in sperm. And this is work that we're really, we're so thankful for Oliver Rando's work in the metabolism, in which he showed that the epididymosomes, which is a part of the testicle in which immature sperm have to pass through, give off a payload of RNA that seem to be linked with what's happening in the liver when it comes to metabolism studies. So we're really focusing on using that framework to see if that can be associated with the traumatic responses.
Okay. Next we have a question from Jin. "Hi, Bianca, that's a very interesting study. Have you studied the effect of oxytocin between male and female mice?"
Yes. Also a question that usually could potentially get me in trouble. So we did study males as well. And what we observed was that males treated with oxytocin seemed to retrieve on the same order as virgins who had never learned from the mom. So we also did the experiment where virgins were treated with oxytocin, but they were not co-housed with the mom. So they never got to learn. And so this was over the span of 7 days. The males treated with oxytocin would begin to respond with retrieval, which in some component we can say that's exciting that males can retrieve. But if we were to look at this from an ecological perspective, taking 7 days to pick up your baby is not ideal in the wild. And so it seems that mice did learn to retrieve, but it was on a longer time period as opposed to the virgins who were treated with oxytocin co-housed on the order of 12 hours.
Okay. All right, so thanks for the great talk, Bianca. I don't think there are any remaining questions, but people can still type in questions for a bit. And oh, here's one from Alex Piet. "Have you tried pairing the pup call with any other neural signal? For example, pup call plus a different neuromodulator?"
A great question. I personally have not tried pairing the pup call with another neuromodulator, but the question is, could another neuromodulator work? Potentially. We're seeing that neuromodulators can regulate excitatory and inhibitory balance. The reason why I focused on oxytocin is because going back to the ecological component, although dopamine may potentially do the same thing, serotonin may potentially lead to this difference in EI balance, in nature, the activation of oxytocin during the birthing process is really what's going to be heightened. So focusing why on oxytocin, because in this experience, the uterine contractions, the milk letdown, and the first salient cue, that's where oxytocin really shines.
Cool. Thanks, Bianca.
Thank you.
Okay, next we have a project talk from the MindScope program put together by the visual behavior team. The visual behavior group will describe how their unique data pipeline has been critical to uncovering how specific cell types contribute to vision and learning behaviors, as well as some of the tools they've built to help others explore these amazing data sets.
Hi, my name is Boaz Lev. Hey everyone, I'm Pete Grobleski, and today, Marina, Doug, Alex, Arena, Farzana, and I are going to talk to you about the Visual Behavior Allen Brain Observatory. So the overarching goal of the Visual Behavior Project is to use large-scale physiology in mice to understand how encoding of sensation and behavior across the thalamocortical visual system is influenced by behavior state, expectation, and experience during an image change detection task. So for this project, we built the Visual Behavior Allen Brain Observatory, which consists of a series of experimental, data collection, and data processing steps shown here. Each of these steps is performed in a standardized way by various teams of technicians. So first, we breed transgenic mice that express a genetically encoded calcium indicator, GCAM, in subpopulations of neurons. We then implant a cranial window in the skull of these mice, which provides us with access to the dorsal surface of the brain. We then use intrinsic signal imaging to map the functional borders of the different visual cortical regions. We then train mice on a visually guided task before the last experimental step of the pipeline, where we image through the cranial window to record from GCAM expressing neurons in specific regions of the visual cortex, while mice are performing the behavioral task. All the pipeline data are then processed and made freely available to the public at brainmap.org. So the major difference between this pipeline and the previous visual coding pipeline is that we want to measure neural responses to behaviorally relevant visual stimuli. So in order to do this, we train mice to perform a simple task wherein they emit a behavioral response, licking a lick spout, right after the identity of a flashed stimulus changes. So it's important to note that with training, these stimuli, in this case, a set of 8 natural images, become relevant to the mice in that they're using the images to guide a learned sensory motor response. So this task, along with the systems that we've built as part of the pipeline, allow us to correlate neural activity, shown here as changes in fluorescence of GCAM expressing cells, with visual stimuli, the flashed images, as well as a set of behavioral signals. And we do this because previous work has shown that visual cortical responding is influenced not only by visual stimuli, but also behavior. So the behavioral parameters that we're monitoring are licking, which in some cases results in reward delivery, running speed, pupil area, as well as face motion energy, which is a measurement of facial movement. So these simultaneously recorded data streams, which are aligned on a frame-by-frame timescale, allow us to address complex questions of how the brain encodes behaviorally relevant visual information during an active behavioral task. So with that, I'll pass it off to Marina.
Okay, thanks, Pete. So the Visual Behavior Project is leveraging the data collection pipeline that Pete just described to ask how sensory and behavioral variables are encoded by excitatory and inhibitory neuron populations. And importantly, how flexible that coding is under different behavioral states and as sensory contexts change. So we are able to target neural populations from 3 major cell classes: excitatory, SST inhibitory, and VIP inhibitory cells, using transgenic mouse lines expressing a genetically encoded calcium indicator. And these populations are of particular interest as VIP and SST neurons are key regulators of the output of excitatory populations. And SST and VIP cells receive diverse top-down inputs and are thought to regulate the flow of information in a state and context-dependent manner, such as during learning. So we've generated a large-scale data set designed to examine the flexibility of representations in these cell populations. And we invite the neuroscience community to share in discovery with us as the data will be publicly released in just a few months in 2021. To test the flexibility of neural representations, we've designed an experiment to measure activity
under varying sensory and behavioral contexts. So first we train mice on the change detection task that Pete described with a set of 8 natural images that become highly familiar over the course of training. And then once mice are performing the task reliably, we transition them to doing behavior under the 2-photon microscope, where we target the same population of cells across multiple imaging sessions. So 3 sessions with the training set, which we refer to as the familiar image set, followed by 3 sessions with a new image set that the mice have not seen prior to 2-photon imaging, which we refer to as the novel image set. So using these 2 image sets, one familiar and one novel, we're able to ask how sensory context, specifically sensory experience, influences cell class specific activity. To evaluate the impact of behavioral context, we interleave the active behavior sessions with passive viewing sessions, in which mice view the stimulus in open loop mode with the lick spout retracted so that no responses or rewards can be given. So in the subsequent slides, we will use the shorthand of F1, F2, F3 to refer to sessions with the familiar image set, and N1, N2, N3 to refer to sessions with the novel image set, where 2 always indicates a passive viewing session. So an additional feature of our experimental design allows us to investigate expectation-related signals by randomly omitting 5% of image presentations from the otherwise highly regular sequence, and doing so only during imaging sessions, but never during training, such that the mice become very accustomed to this repetitive periodic cadence of stimulus presentation, making the rare stimulus omissions an unexpected event. So we can take a look at the overall strength and dynamics of neural activity averaged over the full population of neurons for each cell type for both repeated image presentations indicated by the gray bars and also stimulus omission with the time of the expected stimulus that was omitted indicated here by the dotted line. And we find that excitatory and SST populations are most active during stimulus presentation, and they're relatively silent during omission when no stimulus is present. Whereas the VIP population has very different dynamics, showing ramping activity preceding stimulus onset that dramatically increases in magnitude when a stimulus is omitted. So, of course, this is a very high-level view, averaging across any heterogeneity that exists within the population. And it's only represented here for a single condition, so during active behavior with familiar stimuli. But a key question is how these properties may change as a function of sensory and behavioral context, and whether they're affected by stimulus novelty or task engagement, and how those differences are distributed across the full population of individual neurons. So our experimental design, targeting the same cells across multiple sessions, allows us to examine how the response properties of individual neurons change under different conditions. So here I'm showing an example of a VIP neuron recorded across 6 session types, familiar, novel, active, and passive. And this cell is modulated by sensory experience. So here showing this inner stimulus ramping profile during familiar image sessions, regardless of whether the session is active behavior or passive viewing. So F1 and F3 being active behavior and F2 being passive viewing with the novel or the familiar image set, sorry. And then this cell actually quite dramatically switches its response profile to becoming image responsive during the first session with the novel image set. And then you can see it gradually returns to the omission ramping profile after multiple days of experience with this new image set. So while this is a very interesting example and shows how we can track changes in response properties across days, there's still a lot left to be answered. So this brings us back to our key questions of how sensory and behavioral variables are encoded by these populations. So here we're only looking at stimulus and omission related activity, but animal behavior varies from trial to trial. So could those behavioral fluctuations explain the shift in the response profile? Are there interactions between stimulus and behavioral coding? And how do these interactions change under the different contexts that we've measured here? So addressing these questions requires systematic, integrated analysis that accounts for the full diversity of response profiles and behavioral variability. So in the next upcoming slides, members of the Visual Behavior Project team, Doug, Alex, Irina, and Farzaneh, will be telling you about some analysis that they've done to get at these questions in a more systematic way at both the single cell and population level. So I'll hand it off to Doug. Thanks.
Hi, my name is Doug Olorenshaw. I'm going to talk to you about our modeling efforts to characterize the coding properties of neurons in our behavioral task. Our goal is to systematically characterize how neurons encode sensory and behavioral information in our task, making use of time-varying signals from thousands of neurons and behavioral outputs such as licking, running, pupil diameter, and reward times. One strategy we've employed to synthesize all this information is to build a simple kernel regression model that allows us to predict neural activity. Shown here is the vector y of each cell's activity over time as a function of the task features, such as visual stimuli or licking, multiplied by the weights matrix W. We can then learn the optimal weights to minimize the error between the predicted and actual neural activity. Once we have this model, for each neuron, we can then quantify the contribution of each feature to the cell's response or coding strength by systematically removing each feature or groups of features and asking how much worse the model does. We can also examine the kernel shape or the weights over time to look at neural dynamics and examine how coding strength and dynamics change under different conditions. To build some intuition about how the model works, we can visualize the data streams collected during the experiment, the weights learned by the model, and the model's prediction of neural activity based on those features. What you're going to see here is a short clip where you can see the 2-photon movie of neural activity, the behavior tracking video showing the mouse's movement, the eye tracking video that allows us to measure pupil diameter, the actual neural activity in green and predicted neural activity in white, with visual stimuli indicated as colored vertical bars, the times of licks and rewards, the running speed and pupil area over time, and finally, we can see the contribution of each feature to the model performance at each point in time. Here, one of the 8 images is clearly contributing most to the prediction, with a small additional contribution from our running, pupil, and our hit kernel. This is quantified as the fraction change in variance, explained after removing individual features or groups of features from the model and evaluating how much worse the model does. Here, removing the image kernels individually degrades performance, and removing all of the image kernels together causes the model to lose almost 100% of its ability to fit the neural activity, as shown in the bottom left. Therefore, we can confidently call this neuron a visually responsive neuron. In contrast, we can also look at a cell from our VIP Cree line, which labels inhibitory cells. Here we are occasionally omitting the regularly flashed images, and the cell is active during stimulus emission, and the model is doing a good job of capturing that behavior. When we remove the emission kernel, as shown in the bottom left, the performance falls by nearly 100%. We can therefore call this cell an emission responsive cell. I'll now hand it off to Alex Payette, who will talk about what we can learn from this model across the full population.
Hi, I'm going to tell you about the quantitative metrics we can get out of this regression model. So what do we get out of the model? The first thing we get is what we refer to as the kernel shapes. These tell us how each feature is mapped onto neural activity. For an example, here's the average response of the VIP interneurons to each image emission. Here I'm plotting the normalized change in delta F over F as a function of time. When the image is emitted, the VIP cells tend to increase their activity. However, when we break this apart by what session we're looking at, we find that this is not always true. On the first novel session, where a new image set is being presented, the VIP cells change their activity. Instead of responding to emissions, they tend to have a decrease in activity. Another thing we get out of the model is what we refer to as the neural coding strength. So we can remove a feature or a combination of features, refit the model, and this allows us to measure how important those features were at predicting the cell's activity. I'll tell you more about this in a second. Finally, we can look at functional cell types. We can cluster either the kernel shapes or the coding scores and ask, are there populations of neurons that are encoding the same information together? We could alternatively cluster the features and ask, are there features that are being jointly encoded? So what features are in this model? First, there's visual information, like what image was being presented and was the image emitted. Next, there's behavioral features like did the mouse lick, its pupil diameter, its running speed, and variables we extract from a face video. Finally, there's cognitive information like hits and misses, correct rejects, and false alarms. There's also these behavioral model strategy weights that I haven't told you about. We can group these features together into what we call major and minor components. For example, all the images or all the visual kernels together. All of everything together becomes the full model. What's useful about this nested model approach is we can look at either in a very fine level of resolution or a very coarse level of resolution what the cells are encoding. So how does this work? When we remove a set of features together, we can ask how much did the variance explain decrease? When we look at the visual kernels, we find that across the three cell types, the excitatory, the SST, and the VIP cells, all three cell types are encoding visual information. However, when we remove the behavioral kernels, we see strong differences between the two interneuron types. The SST neurons have relatively little decrease in variance explained, while VIP cells strongly encode behavioral information. In V1, we find relatively little encoding of cognitive information. Next, Irina will tell you about how these coding strengths change over time.
Thank you, Alex, and hi, everyone. My name is Irina, and I'm going to tell you a little bit about things we can learn from the output of our model. The model output allows us to see how neural encoding changes over sessions and task demands. When we look at the variance explained in individual cells across sessions, one of the things that we see is a trade-off between encoding for image stimuli and encoding for running behavior with familiar and novel stimuli. So here we plot the fraction of cells that significantly encode visual stimuli over 6 sessions by cell type. While somatostatin cells strongly encode images both in familiar and novel sessions, we see a notable increase in the percentage of excitatory and VIP cells that begin to encode visual information in the first session with the novel stimuli. When we look at the running encoding across sessions, we see a somewhat different picture. Here we see an increase in the percentage of somatostatin cells that begin to encode running in the first session with the novel stimuli, and a decrease in the percentage of excitatory cells that encodes running, while VIP encodes running similarly across familiar and novel sessions. This suggests that as the task demands change from images seen before to new images, neurons may switch from encoding running information to encoding visual information, or vice versa, and this change depends on the cell type. Instead of looking at variance explained by individual features one at a time or how they are distributed across neurons, we can also see how these features are related to one another. When we correlate features using model output and use hierarchical clustering to visualize their relationship to one another, we can clearly discern 3 categories of features. First one is the visual features with all of our images. The second category is behavioral category with features such as running, face motion, energy, and pupil. And the third category is our cognitive category, which includes features related to the task, like hits and misses. Some things that we see here is that behavioral and cognitive features are more closely linked with each other than one of them with the visual features. We also see that visual features are negatively correlated with the behavioral features. And this means that they typically do not occur within the same cells. This negative correlation can also be seen when we plot the contributions of visual and behavioral features to individual cell activity and how it is distributed across population. On this density plot of dropout scores for excitatory cells, we can see that in the first session with familiar stimuli, most cells are strongly visual, so they are shown in the top left corner. There is a smaller group of cells that is more behavioral in the bottom right corner, and there are some cells that are encoding visual and behavioral information about the same, so they would be in the middle. When we compare this session with the novel session, or the first session where the animals see the novel stimuli, we see that this distribution changes. We still see the cluster of cells that strongly encode visual information, but now we also see emergence of cells that are encoding both visual and behavioral information. So the model allows us to examine how individual cells encode different information across sessions. But another strength of our experimental setup is that we're able to record from multiple cells at the same time, which allows us to look how they work together as a population during the task. So next, Farzaneh is going to tell you a little bit about that. Thank you.
Hello, everyone. I'm Farzaneh Najafi, and I'm going to present results about decoding population activity. The idea here is to see what's the signal at the level of the population. For that, I did linear SVM classification. On each trial, it takes the population activity and predicts some label about that trial. For instance, what image was presented. First, let me show you the cell average responses. In excitatory neurons, neurons on average increase their responses after images. Not much change happens after omissions. In SST neurons, there's a robust response after images, a little bit of decrease after omissions. And in VIP neurons, there is this ramping prior to image representation, which keeps going up after omissions. The decoding analysis that I did is a binary classification. I classified population activity at each time point against the activity right before the omission. What we see is that the excitatory population has high decoding accuracy right after images. This is expected because they have this strong response after images. But surprisingly, there is also high decoding accuracy after omissions. I'm saying surprisingly because, as you see, there is not much going on at the level of the population averages. For the SST population, the decoder accuracy is also similarly high after images and also omissions. For the VIP population, the decoder accuracy is especially high after omissions. So the population decoding results are indicating that all cell types are representing omissions at the level of the population. And they raise a very interesting question, that what is the nature of this signal that the population is carrying after the omissions? In other words, what information is the population carrying? One hypothesis is that the population activity after omissions is carrying information about the subsequent image. To test that hypothesis, I did SVM multi-class classification. And what we see is that in the excitatory and SST population, there is a gradual increase in the decoding accuracy after omissions. Very much unlike the VIP population, in which the decoding accuracy does not go up after the omissions. These results demonstrate that excitatory and SST populations carry image-specific signals after omissions, but the VIP population represents a non-selective signal after omissions. And overall, the population decoding analysis is suggesting an appealing hypothesis, which is different populations in our task are carrying distinct prediction signals about the subsequent image, and their prediction signals are different in terms of their stimulus selectivity, such that VIP neurons are carrying non-image specific prediction signals, but excitatory and SST populations are carrying image-specific prediction signals. This is, of course, a very interesting hypothesis. We seem to have some evidence for it, but there is more to do to test this hypothesis. Thank you for your listening.
Thanks, Farzaneh. So with that, we'd like to thank our founder, Paul Allen, for his vision, encouragement, and support. And we'd also like to thank the entire MindScope and visual behavior team. And we want to give a special thanks to the technicians who have continued to care for our mice and collect data for this project during the COVID pandemic. Thank you.
Thank you, visual behavior team. Super interesting. As a reminder, again, we won't have live Q&A here, so please submit your questions on the forum or come to the visual behavior Q&A session later. So I'd like to introduce our next Gen Leader talk. Our next speaker is Lucas Cheadle. Lucas is an assistant professor of neuroscience at Cold Spring Harbor Laboratory, originally from the Chickasaw Nation in rural Oklahoma. Dr. Cheadle developed a passion for biomedical research as an undergraduate at Smith College. He later earned a PhD in neuroscience working with Dr. Thomas Biederer at Yale University and completed a postdoctoral fellowship in the laboratory of Dr. Michael Greenberg at Harvard Medical School. Throughout his training, Dr. Cheadle's work has focused on understanding how neurons in the brain form precise connections with one another. Currently, Dr. Cheadle's team merges large-scale genomic and transcriptomic approaches, such as single-cell RNA sequencing, with functional assays such as high-resolution imaging of neural connections in the brains of living mice. Using these approaches, the Cheadle Lab characterizes the contributions of non-neuronal brain cells to the sensory experience-dependent remodeling of neural circuits during postnatal brain development. In the future, they hope to turn these insights into novel therapeutic strategies for treating neurodevelopmental disorders. His presentation is titled Non-Neuronal Plasticity and Function in the Stimulated Brain. Welcome, Lucas.
Sorry, was I muted? Thank you so much for that very kind introduction. And I couldn't be more excited to be here and to be a part of the Next Generation Leadership Council. So I really appreciate everybody's attention. Let me see if I can get my slides to progress. From the moment we wake up to the sound of a jarring alarm in the morning, to the moment that we turn the lights off at night, human beings live our lives in constant contact with the world around us. And we perceive the world around us in the form of sensory experience. The ability of the brain to not only interpret sensory experience, but really to mount responses to experience or to make decisions based upon our experiences is a fundamental neurological function, and it's a function that's really paramount to our survival. One of the most important roles of sensory experience in the brain is to promote a developmental process called synaptic refinement. So early on in the assembly of neural circuits, neurons initially form a vast array of connections with many other neuronal partners. But over time, only a subset of these synaptic connections are strengthened and maintained into maturity, while in some cases, the majority of these connections are actually disassembled or eliminated. And I think that process is really beautifully illustrated by these panels of sparsely stained neurons from the human cortex, which demonstrate this robust elaboration of synaptic connectivity that occurs between birth and about 6 years of age. That's followed by this significant pruning down or simplification of connectivity that becomes apparent around the time of adolescence. Now, this process of synaptic refinement, where we remove some of the synapses that we initially make, is really critical for the precise wiring of circuits in the brain. And it's also critical for making sure that those circuits function normally once the brain is mature. But while we know relatively quite a bit about the molecular mechanisms that guide the initial assembly of synapses in the brain, we actually know far less about the fundamental mechanisms through which synapses are eliminated in response to sensory experience. And so my work is really focused on defining those basic mechanisms of neural circuit development. And to do that, we take advantage of the visual circuitry of the mouse as our experimental system. So today I'm going to talk about two visual structures in the brain. The first is the lateral geniculate nucleus, or the LGN, which is a small brain structure pretty much in the center of the brain that receives direct input from the retina, and both processes that input as well as relays that input to the primary visual cortex, which I'm showing here as V1. And the primary visual cortex, as I know many people in this crowd will be familiar, is really the higher order sensory processing domain of the visual system. So both of these structures, I think, are really important for visual function. And the reason why our work takes advantage of these systems is that their connectivity is roughly established by birth, but becomes extensively refined across the first month of postnatal life. And that process of refinement requires visual experience during a temporally segregated stage that spans from about postnatal day P20 to P30, or P35. And so there's a really nice temporally defined phase of experience-dependent refinement in this circuit. And so, I also like to point out that even though vision is a really fascinating aspect of biology, we don't study the visual system because we're particularly interested in understanding vision. We really study it as a unique experimental system that we can use to try to understand the universal principles that guide experience-dependent circuit refinement throughout the brain. So it was as a postdoc that I began to really look into this question of how sensory experience remodels neural circuits during brain development. And in doing this, I formed a hypothesis that would allow me to take an unbiased approach toward trying to identify some of the molecules that are involved in this process. And so the hypothesis that I formulated was that sensory experience, particularly visual experience, may induce the expression of certain genes in relay neurons of the LGN, and those genes may go on to encode molecules with direct roles in synapse refinement. And I think that this is a relatively well-founded hypothesis to begin with, because we've known for several decades that neurons can induce robust gene expression programs in response to neural activity. So we're just taking that observation a step further here and asking whether visual experience can do this in relay cells of the LGN and use those gene programs that we could discover to identify novel molecular pathways regulating this process. And so in order to perform this experiment, I decided to use single cell RNA sequencing. And the reason for that is because around the time when I was planning these experiments, most people were really using whole tissue bulk RNA-seq to investigate gene expression in the brain. But clearly the brain is a remarkably heterogeneous tissue at the level of cell types, as the Allen Institute has been at the forefront of discovering. And so we really wanted to have a way to parse those different contributions of different cells to these gene programs. And so to that end, we actually used a droplet-based strategy of single cell seq, which is called N-drops. And having settled on a technique for looking at gene expression, we next needed a way to robustly stimulate this population of cells in the LGN. And so toward that end, we developed a paradigm. And in this paradigm, we rear mice according to a standard 12-hour light-dark cycle up until the age of about postnatal day 20. And then we rear them in complete darkness 24 hours a day for about a week. At P27, we either harvest their tissue in the dark, which serves as our unstimulated tissue, or we re-expose the mouse to light acutely for a number of hours. And in today's talk, I'll focus on the 8-hour time point. But what this allows us to do is essentially block experience-dependent refinement by placing mice in the dark, and then to look at gene expression at P27, which is really the height of experience-dependent refinement in a stimulated and an unstimulated mouse. And we showed, we validated very carefully that this paradigm really does induce robustly activity-dependent genes in relay cells like FOS, for example, or NPAS4. And so this experiment allowed us to really capture what we're confident in saying are stimulated relay cells and unstimulated relay cells. So single cell RNA sequencing can be looked at in a number of ways, and I don't think that this needs any introduction with the crowd I'm speaking to now, but just very briefly, I'll introduce this plot. It's called a T-SNE plot, and it's a two-dimensional way of representing single cell RNA sequencing data. So in this plot, each of these dots represents a single cell in our data set. That's both stimulated and unstimulated cells. And the distance between any 2 points here is representative of the transcriptional divergence of those cells. So for example, these two dots here, very close together and probably represent the same cell type, whereas this cell here compared to this cell here are very far apart and probably quite transcriptionally divergent. So, so in order to find the relay cells, which are the cells we're particularly interested in, we just looked at marker gene expression and we pulled out these gold-colored clusters as our relay neurons. So now that we'd identified our relay neurons, we could perform a differential gene expression analysis to ask among these cells, are there any transcripts that are more highly expressed in stimulated neurons compared to unstimulated neurons? And the result of that analysis is shown here in the scatter plot, where now each data point represents a single gene across the transcriptome. And the expression of that gene in an unstimulated neuron is on the x-axis, while its expression in a stimulated neuron is on the y. And so what I hope you can appreciate is there's a cluster of red colored dots here that represent those transcripts that were significantly more highly expressed in stimulated relay cells compared to unstimulated. And there's about 213 genes there. And they really encode a number of different functional classes of molecules. So we see some molecules involved in transcriptional regulation, for example. We see some molecules involved in intracellular calcium signaling or even in cell-cell communication. But we decided to home in on one of these genes in particular. And for that, we actually focused on the single most highly induced gene in this data set, which was called FN14. FN14 is a tumor necrosis factor receptor superfamily member. So it's a cytokine receptor. It's a cell surface receptor, so it's localized to the surface of the cell. And we found it to be sort of an interesting or an intriguing candidate regulator of experience-dependent refinement, just based on the fact that it has well-characterized roles in the periphery in the context of tissue remodeling after injury and in the context of immunity, but its expression and function in the brain had not really been looked at. And so we thought that this could be an interesting direction to go in. And of course, as a cell surface receptor, it's very interesting to think that there may be a ligand out there in the brain potentially collaborating with FN14 to drive this process of experience-dependent refinement. So we were really curious first to discover whether the ligand of FN14, which is a cytokine called TWEAK, which is a tumor necrosis factor cytokine. So you can think of it as a sibling to TNF-alpha. We were curious to know what TWEAK expression looks like in the brain during this time as well. And so this is really another plug for big unbiased transcriptomic data sets and how valuable they can be to the field. So we had actually published a single cell RNA sequencing resource where we just looked at the cell types in the lateral geniculate nucleus across postnatal development. And because we had developed that resource, we were able to go back in there and ask about a specific gene and what its expression pattern was in the developing cortex. So we looked at TWEAK expression. So what I hope you can appreciate here is that prior to the onset of sensory experience, which occurs at about P14 when the eyes open, TWEAK expression is relatively low and ubiquitously expressed across a number of different cell types. But right around the time of eye opening, so between P10 and P16, we see this really drastic upregulation of TWEAK expression, specifically in microglia. And we find that this expression actually increases in microglia right into the phase of experience-dependent refinement in the LGM. And as I'll mention later, I'm very much trained as a classical neuroscientist. So previously, the way I thought about synapses and synaptic connectivity was really as a two-component system, a presynapse and a postsynapse. So this idea that this ligand that might be involved in this process is being turned on in microglia was really something that got my attention. And so I sort of learned at that point, what all these non-neuronal cells were doing in the brain. And microglia are interesting because they're the predominant cell type in the brain that's a non-neuronal cell type that is not derived from a neural lineage. So microglia are derived from myeloid progenitors in the yolk sac. They actually colonize the brain during embryonic development. And once they get into the brain, they play really important roles as the innate immune system of the brain. So they respond really quickly to brain injury. They also respond to pathology. But work over the past 12 years or so have started to define a host of different types of functions for microglia in the healthy brain. And so we really thought this was an interesting finding, and we're intrigued by the idea that perhaps microglia are actually signaling to neurons to help refine the circuit in response to experience. But the first question I wanted to ask was, I was very intrigued by this induction in microglia occurring right around the time of eye opening. And that made me wonder whether visual experience could be inducing gene programs in cell types in the brain other than neurons. And so to look at this particular gene specifically, we returned to our visual stimulation paradigm. And this time we performed this paradigm in harvested tissue and performed fluorescence in situ hybridization. And so this is a confocal image where I'm showing you two different microglial cells here, which we've probed for with the probe detecting the gene encoding the receptor, fractalkine receptor, which is a microglia specific gene. And then we're looking at tweak expression in green and FN14 expression in blue. And so in the left-hand column, you're looking at a microglial cell from an unstimulated mouse. And I hope you can appreciate that there's essentially no expression of tweak or FN14 in this context. But now if we look in a mouse that's been acutely re-exposed to light for about 8 hours, we see that there's actually this robust induction of tweak expression. But because we're looking at microglia, we don't see FN14 at either time point. And FN14 happens to be neuron specific. So we can turn the page and actually look at neurons instead. And now we can validate that tweak expression is actually definitely not happening in neurons, but that FN14 is being induced in this completely different cell type. And of course, we can quantify these results, and I'm showing those quantifications here in the form of cumulative frequency distribution plots, where the x-axis on the left represents the number of tweak molecules per microglial cell, and the on the right represents the number of FN14 molecules per neuron. And I hope that you can appreciate these drastic rightward shifts of the cumulative frequency distribution in both microglia and in neurons. And so this really highlights two different things that I think are really interesting about the data. The first aspect is that non-neuronal cells, even cells that are derived from a completely non-neural lineage, can mount transcriptional responses to environmental stimuli, in this case, visual experience. But the other factor is that because both of these signaling partners are being induced in completely different cell types, I think it really hints at maybe a higher level of control or organization that experience plays over the way that different cells communicate in the brain. And in particular, it looks like those interactions that are being controlled by experience may have really critical roles in shaping synaptic connectivity. And so because this work is already published, I want to just summarize on the next slide the electrophysiological and ultrastructural results that showed us what tweak and FN14 actually do in the context of sensory-dependent refinement. So we find that upon the onset of sensory experience around eye-opening, FN14 expression is localized to synapses, and it serves to increase the number of dendritic spines and the number of synapses per dendritic spine, so both parameters, in response to experience. And we think that this serves to strengthen and maintain some of these retinogeniculate connections. But at synapses that happen to be particularly nearby microglia that express tweak, we find that tweak binds FN14, disassembles dendritic spines, and ultimately leads to the elimination of synapses. And now I told you before that it's really this balance between synapse strengthening and synapse maintenance and synapse elimination that defines this process of synaptic refinement that's coordinated by sensory experience. And so this pathway really provides an intriguing example of how a single signaling pathway can sort of balance both of these two features. And I really think that's important because it starts to address this key question in synaptic biology. It's been, how does a protein in a single neuron go on to regulate some synapses so that they're strengthened and others so that they're eliminated? So in the context of our model, we suggest that this feature is being conferred to some extent by extracellular cues coming from glia. Now, as I mentioned, I'm classically trained as a neuroscientist, and I've always been interested in synapse development and synapse refinement, but canonically, the thought process in my mind has really looked like this, this sort of example I'm showing you here, where I think about the brain as really a connection between different neurons. And I think about visual experience as inducing these interesting changes at synapses. So in this particular example, I'm showing you a synapse being strengthened and a synapse being eliminated. But having made this discovery about microglia, it's really sort of shaped the way I think about this. And now I realize that the brain is much more than just a series of interconnected neurons. The brain is really packed with different cell types that are playing very active roles in remodeling the circuitry, the synapses of the brain, and maintaining different aspects of homeostasis. And when you have a visual stimulation, it's now very clear that that visual stimulation impacts glia as well, and that these glia are actually both being plastic in response to these cues, but also those changes in non-neuronal cells are in some ways organizing the changes in synapses. And so that's something that I find very interesting, and it really became the basis of my own laboratory, which I set up 4 months ago at Cold Spring Harbor Lab. And so in the next few slides, I'd just like to show you some unpublished, very new results to give you a sense for where my laboratory is going. So being someone who really values unbiased transcriptome-wide approaches, we wanted to begin by taking an unbiased approach to really characterize non-neuronal gene expression that is caused by visual stimulation. And so this is a project that is being driven by Johan August in my lab, who is a research technician who was the first person in my lab. And what we've done is we've returned to our visual stimulation paradigm. This time we're not in the LGN. This time we're going into the primary visual cortex. There are a number of reasons why that's a great system to do this in that I won't get into because I think they're pretty clear to folks at the Allen, but it's a wonderful system. It can be stimulated with the same paradigm, the visual stimulation paradigm, and we get a lot more tissue out of the V1 than the LGN, so it's helpful in that way. So we microdissect out V1 from stimulated and unstimulated mice, and now we subject the tissue to a variety of optimized enzymatic preparations as well as temperatures. And the goal of that was to optimize for non-neuronal captures. So most of these studies really focus on neurons, but we're trying to sort of reverse engineer that. So we developed a single cell dissociation protocol that really captures those non-neuronal cells. And we submit that to single cell transcriptomics, and we can then identify transcripts that are differentially expressed in response to visual stimulation. And so this approach of sort of optimizing that single cell dissociation protocol did allow us to capture a predominance of non-neuronal cell types in the brain, including a variety of microglia and astrocytes, as well as oligodendrocytes and vascular cells. And we still get neurons, and that's important because we're ultimately interested in how neurons and non-neuronal cells communicate. We're not just interested in one particular side of that interaction, really interested in all sides. And you can also see the diversity of our cell types through this UMAP, which is similar to the t-SNE I showed earlier. But I think it gives you a nice sense of the cellular repertoire that's represented in our data set. And so now we're really interested in taking a step back and asking, are there any transcripts that are significantly different in stimulated non-neuronal cells versus unstimulated? And as I'm showing here in this plot, which is called a strip chart, every cell type in our data set induced gene expression programs in response to visual stimulation. So the y-axis represents the log2 fold change of stimulated expression over unstimulated. So each of the colored circles in the data set above the 0 represent those genes that are significantly upregulated in an experience-dependent manner, and those that are below the 0 represent those downregulated genes. But for our purposes, we really decided to focus on the upregulated genes, at least in the beginning, as potential candidate regulators of experience-dependent refinement. And this is really a process that we're still just jumping into, trying to understand whether there are any interesting functional ramifications with these cell types. But something that I will say that I think is really important and really telling is that these inducible gene expression programs look to be very cell type specific. And that's consistent with two things: A, cell type specific functions being driven by experience, but B, it's also consistent with potentially each of these cell types having a unique epigenetic or transcriptional program that drives these different gene expression responses. And so while I can't go into too much detail yet on the functional aspects here, I will say that a number of interesting findings has jumped out, including the presynaptic protein orexin-1 being upregulated in microglia. And something that I'll mention, because I found the talk earlier on the EM data looking at the oligodendrocyte precursor progenitor cells to be absolutely fascinating. I actually think that's a really, really interesting cell type in the brain. And I think that they're underrated in terms of what they do, because people just assume based on the name, they're just waiting to become oligodendrocytes. But I actually think they're their own functional class. And I think that the data I saw today certainly supports that. But one of the things we noticed is that we actually see an interaction, potential interaction between microglia and OPCs in our data set that may be driven by protein tyrosine phosphatase signaling. So definitely be interested in learning more about what they're doing in terms of phagocytosis. But I think that's the type of power that a data set like this has. It allows you to go in and make hypotheses about how cell types connect to one another. But in my laboratory, we're really ultimately interested in going from transcriptional programs to cellular function. So we're not solely wanting to do bioinformatics. We want to really get to a functional readout. And so one of the things we've been doing to connect the transcriptional data to functional data is to look at microglia in tissue and to combine fluorescence in CT with protein immunostaining, so that within a single microglial cell, we can see how much of a given gene it expresses, and we can see how many synapses it engulfs. And this is just an example of what such data look like. It's a 3D reconstruction of a microglial cell in green that's transparent so that you can visualize the thalamocortical synapses in blue, which are stained for VGLUT2. And in red, you're seeing the RNA that expresses C1qa, which is a protein that's important for phagocytic engulfment of synapses, and that we've also found to be upregulated by experience. So we get really beautiful single cell data from this assay, and then we can quantify changes in RNA expression as well as synaptic engulfment, such as in this graph I'm showing you here. And in this graph, the 0 time point just means dark-reared mice, so unstimulated. And each tick after 0 is amount of time that the mouse is re-exposed to light. So I hope that you can appreciate, we see this interesting peak in synaptic engulfment occurring right around 6 hours post-stimulation that then is completely returned to normal by 10 hours. And we see somewhat of a reciprocal effect in C1qa expression, where it dips down at 6 hours and then increases at 10. So we think that this correlation is starting to give us a hint that these gene programs are really important for the ability of microglia to participate in visual circuit plasticity. And so my lab is really taking two directions off of these data. And one is very genomic, transcriptomic focused, and the other is functional. So as I mentioned earlier, I think that the fact that these gene programs in non-neuronal cells are largely cell type specific, hence not only that they're doing different things in response to experience, but that the nuts and bolts machinery in the nucleus that organizes these gene programs probably are to some extent distinct. And so the first thing we want to do is really define those genomic mechanisms in microglia particularly. And to do that, we're using a transgenic mouse line to express a GFP tagged nuclear protein, SUN1, selectively in microglia. And I'm showing you the validation of that approach here with our GFP tagged nuclei in this panel and our microglia labeled in this panel. And what we're doing is we're using this approach to acutely isolate microglia from the brains, particularly the visual cortex of mice following dark rearing and a number of different time points after light re-exposure. And we're doing attack sequencing to look at chromatin accessibility. We're doing RNA sequencing to get a deeper dive into their transcriptional programs. And ultimately, we're interested in doing other approaches like cut and run to really try to define what that transcriptional machinery is in microglia that's driving these programs. And the other side of the lab, we're actually building a 2-photon microscope so that we can actually go into the visual cortex of living, awake mice and measure microglial responses to the visual stimulation paradigm that I've introduced. And we want to measure those responses at the level of structural interactions between microglia and synapses, particularly thalamocortical synapses. But additionally, we're interested in using genetically encoded calcium sensors like GCAMP to actually measure calcium dynamics in microglia that occur in response to visual stimulation. And while it's really early days for doing this and for understanding what these calcium transients mean in microglia, there's a really exciting paper that just came out showing that these calcium transients are really tightly attuned to neural activity. So we think by doing some interesting work in visual cortex, we'll be able to harness these approaches to really try to link those transcriptional changes we're seeing in microglia to functional changes in synaptic connectivity. And with that, I'd like to thank the people that were instrumental in this work. I showed quite a bit of work from my postdoctoral fellowship. So I first want to thank my postdoctoral advisor, Michael Greenberg, first and foremost, who's been a fantastic mentor and a very generous mentor as well. And I highlight three other people who were specifically involved in this work: Chris Zhang, Brian Kalish, and Sam Rivera. as well as a number of collaborators. And of course, I want to end by thanking the people in my own lab, Johan August, Uma Vrudhula, and Austin Farrow, along with a host of other folks at Cold Spring Harbor who have just been absolutely instrumental in getting this research program off the ground. And finally, I want to thank our funding sources, the National Institute of Mental Health and the Brain Initiative. With that, I'm happy to take any questions.
Hey, thanks, Lucas. Please submit your questions for Lucas in the Q&A. We do have one question so far, and this is from Anatoly Buchin. How did you determine cell types? Would it be possible to connect them to the types identified in Tashik et al. 2018?
Yes, definitely. So we haven't done that as rigorously as we could yet, but we use the same types of marker genes. So I think the marker genes are really well established at this point. So I think it's as simple as just making sure that you're looking at, clusters that have the enriched genes in them. And those genes to a large extent are derived from data sets like that of Tashik. So the only thing we haven't done that I do think would be interesting though, is because those data sets like the Tashik data set do also have information about non-neuronal cells. One of the things we're really interested in doing is actually converging what we're seeing with some of those other data sets from the Allen Institute, particularly, just to see if we can dig a little bit deeper in, but also just to validate our cell type designation.
Okay. This is one of the first studies to show that microglia are important in these processes. And if not, are there similar mechanisms at play in other cortical areas that you know of?
Yeah, so microglia, there was a discovery made about 10 to 12 years ago. Actually, 2011, I think, was the first time it was really explicitly shown that microglia contribute to neural circuit development by pruning synapses, and that they do that by phagocytosing or engulfing synapses. And so this was shown in hippocampus as well as in the LGN. But the twist is that microglia actually stop engulfing. They engulf many fewer synapses during these phases of development that I'm talking about, compared to those earlier phases, which was really the base of the Ben Barres, Beth Stevens sort of college of discovering these roles. And so that's something that's really interesting about the tweak FN14 data I showed you in the LGN. We actually think that that occurs through a non-phagocytic mechanism. We actually think that occurs through a tweak binds FN14 and it leads to this neuronal deconstruction of the synapse. So it's a little bit different of a mechanism. But in the visual cortex, on the other hand, as you saw at the end of my talk, we actually do see that engulfment is involved in the pruning of those synapses. And I do think that we're one of the first studies to show in a developmental context that microglia disassemble synapses in response to experience, but we're certainly not the only people to show this. And there was a paper in 2019 that was a collaboration between Dory Schaefer and myself, actually. I was on the paper where we showed that this is happening in somatosensory cortex at a much younger age. And so we showed that. And then there was also, there have been a couple of other studies more recently. For example, there's a recent Cell paper showing that microglia can participate in hippocampal plasticity by phagocytosing extracellular matrix. So there's definitely a couple of examples of papers that really lend support to this idea, but it hasn't really been systematically investigated. And that's one of the things that we want to do in the lab, is really to systematically define the molecular mechanisms to which microglia remodel synapses.
Awesome. Okay, so we have another question from Forrest Coleman. Forrest says, thank you, Lucas. I'm curious whether you think there are cell type specific gene expression changes in non-neuronal cells that are specific to different cell type specific remodeling. Say most bluntly, remodeling of excitatory versus inhibitory networks.
So I guess the question is, when a microglia sort of expresses all these different genes in response to sensory experience, do some of them encode regulators of inhibitory versus excitatory synapses? My thinking would be yes. I think we don't know as much about how microglia remodel inhibitory synapses versus excitatory, although I know that there's a lot of work that's about to come out on that topic. But from what I've seen, it does look like they play different roles with inhibitory versus excitatory synapses, which would certainly hint that if their transcriptional programs are contributing to both of those, they would be distinct subsets of molecules that might be important for each. Like for example, inhibition, inhibitory synapses, actually it's been shown in the cerebellum, microglia actually like induce the integration of inhibition. So I think there's definitely some synapse specific variability, and it would be my hypothesis that some of that derives from experience dependent transcription.
Cool. Okay, so the next question is one I also had. I'm really interested to find out. By Peter Burbach, he asks, how do the microglia perceive the light stimulus?
Yeah, so this is actually a major sort of theme in my job proposal, essentially. So I think there's a number of different possibilities. So the one that I sort of champion as my first hypothesis is that, we know that when neurons mount transcriptional responses to sensory experience because they're synaptically active, right? So that makes sense. A lot of the genes that they actually induce in response to that cue happen to encode secreted molecules as well as cell surface molecules. So one hypothesis is that neurons are the first to respond to these stimulus-dependent gene programs, and then they secrete or release factors that recruit non-neuronal cells. So that's one possibility. The other possibility is that microglia express neurotransmitter receptors. So it's possible that because they're so intimately contacting synapses all the time, they could actually detect some fluctuations in neurotransmission as well. Other possibilities include the idea that the neurovasculature changes when you have a robust stimulus. So it could be something to do with blood flow or oxygen in the tissue. I think there were a lot of hypotheses, but I think that by defining the gene programs in microglia, so for example, one of the things we're doing with our data set is parsing out the ligands and the receptors. So what are the genes that are changing that could be participating in these different cell-cell interactions, and then looking at where those ligands and receptors are expressed. So taking a bit of an interactomic approach to it. But I think it's one of the most fundamental questions about what we're doing. And so hopefully we'll be able to derive some insight into that.
Thanks. Okay, Ben Slivka wants to know, the count of cell types keep increasing. It seems like we are still in the zoology era of cell typing. Even though we can do gene sequencing, what could change that?
What could change the...
I'm guessing the fact that the cell type numbers keep increasing. I think it's your question to answer.
No, I'm just not sure. I just trying to make sure I understand. So you mean like the glial cell types are increasing? Or?
I think it sounds like just cell types in general. The more we look into it, this is something that I deal with too. Like the more we look into cell types, the more, it's sort of a philosophical question, I'm guessing. Like what might stop the increasing count of cell types that we seem to be getting?
So my first answer to that is that I'm actually a little bit less on the taxonomic side of this. I actually like to use single cell RNA sequencing to compare conditions more so than I like to piece apart like how many types of microglia there are or astrocytes or whatever. So I try to let other people do that. It's my personal opinion that cell type definitions for me are most valuable when we think about them in terms of function. So does it matter that two cells are slightly different transcriptionally if they both do the same things and act the same way? I mean, I think the hypothesis for a lot of folks that do these taxonomic investigations is that if they're different transcriptionally, I mean, it's quite likely that they have different functions, at least subtly. I mean, it might be that we don't have the precision in terms of tools to necessarily show how different they are. And I think that's likely, and I think there's a lot of value to that. But to stay out of the weeds with that, I tend to try to focus on categorizations that I'm very confident in, like fractalkine receptor expressing microglia. But I think one of the big questions in the field right now is, are there functional subtypes of microglia? And so I do think it's like a very timely question to be asking.
Yeah. It's pretty interesting because you could conceive of a time when cell types are defined by functionality and then transcriptomic types are lumped in together on that basis, if I read you right. Okay.
Yeah. Yep.
So the next question is from Zhizhen Yao. She wants to know, is FN14/tweak associated with any particular neuronal types?
Yes, and it depends on the brain region. So in the LGN, for example, there's really only two neuronal types in the LGN, though. So it's a little bit of a limited system, but it's in excitatory but not inhibitory. LGN, but its expression pattern seems to be different in different parts of the brain. For example, in the cortex, it's actually quite highly expressed in endothelial cells, FN14 is. And so it really varies by brain region. In the LGN, it's excitatory. So that's really interesting. But yeah, we haven't so much explored farther beyond that.
Okay, our next question is from Maria Pompiano. She wants to know, just a detail, do you have any indication that CFOS is expressed by non-neuronal cells like microglia as a result of visual experience? Thank you. Very interesting talk.
Yeah, so I think so CFOS does not come out as significantly induced in microglia in my particular data set. But CFOS is actually induced in so the Greenberg lab had an adult cortex paper come out, single cell stimulus dependent stuff in adult, and they had seen FOS turning on in a variety of non-neuronal cell types, including endothelial cells, which is something we've also seen, as well as other types of cells. So FOS and its family are certainly by no means neuronal specific transcriptional regulators. I mean, they're very broadly expressed cancer and other tissues, and they're very stimulus dependent, whether you're putting neuroactivity in or whether you're putting any other thing in. So I think that it's, yeah, so we haven't really seen it ourselves in microglia, but I wouldn't be surprised whatsoever if we did like a more targeted approach, like maybe in situ hybridization if we were to see it. But I think the question is relevant to what we're doing since we're interested in those transcriptional mechanisms in microglia. But I will say that I do have a hypothesis that some of the transcriptional regulation is going to be unique to microglia. And so hopefully we'll have some answers to that soon, but it's a really relevant question though.
Okay, our next question is from Daniel Felleman. He wants to know, does the LGN remodeling occur equally across retinotopy? In some species, central vision relay will be pruned to one retinal ganglion cell input per LGN relay. Does that occur in mice and in the periphery?
We didn't actually look at any particular part of the LGN when we were studying this. We made a decision to look across the different regions, although I think that, you know, now that we have the precision and the knowledge that there is sort of this not only retinotopy, but other aspects of organization to the LGN, like shell versus core, ipsilateral versus contralateral, I think that I think that it's a great experiment to do in the future, but it's not one that we didn't pay much attention to that aspect in our studies. In terms of the connectivity of LGN neurons, there was a paper a couple of years ago that did some viral tracing from LGN neurons and asked, you know, what retinal cells were connecting. And I think they found a surprising variety of different configurations, everything from monosynaptic connections to a retinal ganglion cell that's connected to up to 90 different, a relay neuron that's connected up to 90 different retinal cells. So I think it's a really interesting system, but we didn't look at those substructural differences.
Okay, we have a question from Kathy Murphy. Are microglia spitting out tweak and FN14 expression is limiting which synapses are removed, or is there some localization specificity to the microglia that contributes to which synapses are removed?
The latter is, it could be both, but the latter is what we've actually shown. And so what we found, and I didn't bring this up, but when we talk about tweak being induced in microglia, it's actually not induced in every single microglial cell. It's actually induced in most microglia, but not all. So what we found is that when a microglial cell does express tweak, there tend to be fewer synapses nearby that cell compared to microglial cells that don't. And we think that argues for what you're describing, essentially a proximity-based model where there's synapses near that tweak-inducing microglia that happen to be more likely to be removed versus others. But at the same time, you want to have some specificity, right? You want some functional specificity. So we're not exactly sure beyond that how those synapses are being chosen, which are strengthened and which are eliminated. But it certainly has to do with the proximity of them to the microglia that expresses tweak.
Okay, thanks Lucas for a great talk. Next up, we have the Human Cell Types Project talk. It's our final session today. Let's see, This team will cover our latest work to uncover conserved and divergent cell types across mammalian species, specifically human.
Hi, my name is Boaz Levy. I'm an assistant investigator, today representing the Human Genetic Tools group and the CNS gene therapy teams that I co-lead with Jonathan Ting, who is not going to be presenting today, but his work is critical for all the slides that we will be showing. The Human Genetic Tools group stands on the shoulders of really fantastic work that has been done by the transcriptomics groups at the Allen Institute, where both human and mouse cortex has been profiled extensively through single-cell transcriptomic studies. We can see good correlation between the cell types between mouse and human, and by arraying them in a taxonomy-like structure, such as this, we can clearly see cell classes, such as non-neuronal classes and glutamatergic cell classes, subclasses such as the parvalbumin subclass, and these final leaf nodes on these taxonomies that are cell types. The goal in the Human Genetic Tools group is to build new AAV viral vectors to allow us to gain genetic access to these cell classes, subclasses, and types. The way we do this is harnessing the power of enhancers. Enhancers can be revealed from human tissue by isolating nuclei and then subjecting them to single nuclear attack-seq. This is a technique that reads out regions of open chromatin. We've done this on the middle temporal gyrus of the human, and our first initial data set showed clear resolution of cell subclasses. We can look into this data and we can find peak pileups. These are regions of open chromatin that are putative enhancers. And many of these are only accessible in discrete cell subclasses. So the way we go about building these viral vectors, first we prioritize the cell subclass or type. Then we go find regions of open chromatin that are putative enhancers that we would like to test to see if they can drive gene expression in these cell classes. We clone them and put them into an AAV viral vector to drive a reporter expression. And then we test them in a combination of species such as mouse, macaque, and in human slice culture. And over the last several years, this has been a really successful effort. We've tested a number of these in both mouse as well as primate. And we've identified enhancer AAVs that give us a variety of cell class and subclass specificities, such as astrocytes, shown here in mouse and in non-human primate. We have enhancer AAVs for oligodendrocytes, as well as calretinin interneurons, and layer 5 extratelencephalic cells. However, it's critical that we actually test that these function across species, because sometimes we identify enhancers that give us very clean selective expression, such as this enhancer AAV that drives expression in layer 5 ET cells. But when we tested it in primate, it gave much broader expression profiles. So today, I would like to hand the mic over to these 4 excellent scientists who will tell you short vignettes about work that's being done in the Human Genetic Tools group, as well as the CNS gene therapy team. Yi Ding will tell you about how she's beginning to really ramp up our single nuclear attack-seq studies, which we hope will allow us to identify cell type-specific enhancers. John Mish will talk about a collection of parvalbumin interneuron-specific enhancer AAVs. Brian Gore is going to tell you about how we hope to use these AAV vectors to deliver therapeutic transgenes to help cure neurological diseases. And last, Natalie Weed will finish the section by describing a really exciting new collection of striatal-specific enhancers. So with that, I'd like to introduce Yi Ding, who will tell you about single nuclear attack-seq.
Hello, everyone. My name is Yi Ding, and I'm a scientist in Boaz Levy's team. Today, I'm going to talk about cell type resolution single nucleus ataxic enables enhancer discovery. Chromatin accessibility is dynamic across the genome, and open chromatin regions enrich for regulatory elements. An assay called ataxic can be used to study open chromatin regions in the genome. Ataxic uses a hyperactive TN5 transposase to insert Illumina sequencing adapters into accessible chromatin regions. Single-cell ataxic assay provides similar resolution to conventional bulk ataxic with additional information in cell-to-cell variation. We are currently using the single-cell ataxic assay from 10x Genomics. In this approach, nuclei are first isolated from a single-cell suspension and transposed in bulk with the transposase TN5. Transposed nuclei are then loaded onto the microfluid chip for gel bed in emulsion generation. Each gel bed is functionalized with single-stranded barcoded oligonucleotides to incorporate barcodes to transposed DNA. Approximately 100,000 germs are formed in each channel, resulting in the encapsulation of tens of thousands of nuclei in germs per experiment. After germ generation, barcoded DNA is poured for PCR amplification to generate index libraries for high throughput sequencing. In the past, we have looked for cell subclasses because that's the resolution we had. One of the reasons we are doing high throughput method is because we want to be able to identify cell type specific peaks. To find distinguishing neurocortical cell-specific peaks, we generated high-quality chromatin accessibility profiles from multiple postmortem human MTG regions using the 10x single-cell ataxic assay. The donors are aged but neurologically normal with short postmortem interval. After strict quality control filtering and mapping to human single-nucleus RNA-seq cell types, we get a data set of over 37,000 nuclei. We carried out iterative clustering and mapping to Rebecca's RNA-seq dataset. In the first iteration, we grouped nuclei into 15 subclasses based on MTG subclasses. In the second iteration, we grouped nuclei into 3 main cell classes: GABAergic neurons, glutamatergic neurons, and non-neurons, based on their subclasses identified in the first iteration and marker gene accessibility score of each cluster. For each main cell class, we performed another round of clustering and mapping to identify major cell types. In those UMaps, different colors represent different subclasses or cell types. In total, we found 43 GABAergic cell types, 21 glutamatergic cell types, and 6 glial cell types, which is almost all of the cell types identified through our transcriptomic analysis. And this is exciting, and it shows that we have cell type resolution from our ataxic data. Using these automatic tools, we are able to identify cell type specific peaks for some of their cell types, but not all of them. On the left panel is a heat map of marker peaks with color indicating the z-score of log2 normalized accessibility. An expanded dataset may allow identification of peaks in all cell types, or it might not be possible for some of them. Since we see a pile of reads right here in this putative enhancer in this layer 5 NP cell type, but none of other cell types, it looks like this could be a cell type specific enhancer for layer 5 NP cells. In conclusion, cell type specific enhancer could be used to generate cell type specific AAV tools, and Joan Mesh in our group will talk more about generating specific enhancer AAV vectors.
Thank you, E. My name is John Mish, and I'll tell you about a collection of parvalbumin interneuron-specific enhancer AAV vectors. We focused on identifying enhancer AAV vectors to target parvalbumin-expressing interneurons because these cells are very important to regulating the brain microcircuit, as evidenced by their dysfunction in diseases like severe epilepsies and perhaps also Alzheimer's. So we set off to identify candidate enhancers that we could use to target these cells. We found 20 different candidate putative enhancers from the single nucleus ataxic data shown here that were generated from the data like E showed you in human. Some, but all of these were also detected in similar mouse single nucleus ataxic data sets. When we looked at the accessibility profiles of these putative candidate enhancers, we found that there was a diversity of them, with some having weak specificity for parvalbumin expressing interneurons, some having very specific accessibility profiles only in parvalbumin expressing interneurons. When we actually went and cloned all 20 of these out, we found that overall 7 out of 20, or 35% of them, did show some levels of parvalbumin subclass specificity, from ones with weak specificity in dark green to ones with very high specificity in light green. 2 of those are shown here. On the left, EHET023H shows low levels of specificity, from 47 to 66% of the cells that were labeled by the vector are parvalbumin expressing interneurons in V1 of cortex by both multiplex FISH, HCR, and by single cell attack RNA-seq studies. And in contrast to that, EHET140H has very high levels of specificity, from 98 to 99% of the cells having parvalbumin expressing interneuron identity by multiplex FISH and single cell RNA-seq. So it's clear that we can isolate a variety of different enhancer AAV vectors that target parvalbumin interneurons. And furthermore, when we looked at the whole brain expression patterns of them, we saw that some of them, like EHET023H, labeled many of the sites of expression throughout the brain that are also seen by the endogenous parvalbumin transcripts, like not only neocortical interneurons, but also Purkinje cells in the cerebellum and nuclei in the midbrain and hindbrain. In contrast to that, EHET140H specifically only labels neocortical interneurons. So this suggests that that our collection of parvalbumin expressing interneuron and enhancer AAV vectors have a variety of different use cases and a variety of different properties that we can use for various biological or eventual therapeutic studies. Furthermore, we wanted to validate that they're not just active in mouse only, that they can also work across species. So we injected them in multi-site in vivo injections in living primate. We were able to harvest the brain tissue and find the expressing cells by anti-SYFP2 immunohistochemistry and co-labeling with parvalbumin parvalbumin molecule reactive antibodies. And so the green dots show the high level of specific on-target labeling of SYFP2 expressing cells that also express parvalbumin. 4 of our top enhancers that we tested in this assay do show high levels of specificity, from 86 to upwards of 98% of the cells that are labeled by these vectors are parvalbumin expressing interneurons in the neocortex. And furthermore, we were able to achieve very high completeness levels. Almost 90% of the parvalbumin expressing cells can be labeled by these vectors in certain cases. So we think that these will be a very useful tool to studying the biology of these cells. Furthermore, we took one of the enhancers and we labeled it across in multiple regions: occipital, temporal, somatosensory, and motor cortex. We saw that across all 4 regions, specificity maintained high levels of specificity, as well as completeness remained high as well. So we think that these really are useful tools for labeling parvalbumin cells across the monkey brain. Finally, we were able to use these tools for direct in vivo characterization of the cell's properties, so that we could target them for electrophysiological studies. We'd see that the cells that were labeled by the EHET140H vector, 100% of the labeled cells show high gain and low AP half-splits, consistent with them having fast spiking interneuron electrophysiology which is consistent with their parvalbumin molecular nature. So to conclude this, I would like to say that we have generated a collection of enhancer AAV vectors that target parvalbumin inhibitory neurons in the brain and with differing sets of properties that we can exploit. These vectors do show conserved activity from mouse to primate and enable cell type specific functional experimentation across species. And we think that these tools will be useful elements to target p-value cells across species for either research or clinical application. In the next segment, Brian Gore is going to tell you a little bit about our plans for using tools like these for eventual clinical therapeutic applications. Thank you.
Hello, I'm Brian Gore, and I'm on the Human Genetic Tools team. We are developing adeno-associated viruses, also known as AAVs, to generate gene therapies for various neurological diseases. Around the world, several hundred AAV gene therapy trials are being conducted and giving hope to patients burdened with life-threatening diseases. AAVs are a safe and low immunogenic viral vector that enable the effective delivery and expression of transgenes to diverse cell types throughout the brain. Shown here in the upper left is administration of an AAV to the brain ventricular system of a mouse at postnatal day 2. By delivering genes early in life, we hope to stop disease progression for severe neurodevelopmental diseases. This AAV had contained the human synapsin promoter that drove widespread viral transgene expression throughout the brain. For many gene therapy disease indications, however, widespread expression could raise safety concerns of potential side effects from off-target gene expression. We know that both genes and diseases demonstrate a high degree of cell type specificity. GABA inhibitory neurons, shown on the left, are found throughout the brain. And despite only accounting for 20% of the total neuronal population, their dysfunction can cause certain mental disorders such as autism, intellectual disability, and deadly forms of epilepsy. To advance therapies to the clinic to correct these disorders, we need better gene therapy viral vectors that only express in the correct cell type. The importance of conferring cell type specificity also extends to neurodegenerative diseases. Shown in the middle are the striatal medium spiny neurons that degenerate in Huntington's disease. And on the right, the midbrain dopamine neurons that die in Parkinson's disease. If we are to treat these diseases in a safe and effective manner, we must discover and develop genetic regulatory elements that precisely control therapeutic transgene expression. Our goal is to develop compact AAV enhancers that drive specific expression in every cell type of the brain. We hope these tools will prove valuable for both basic neuroscience research, but also advance the next wave of gene therapies. We have generated a diverse library of hundreds of AAV enhancers that restrict gene expression to subsets of brain cell types. Here are a few examples that show AAV-based expression that include Purkinje cells of the cerebellum, astrocytes, median spiny neurons of the striatum, and oligodendrocytes. Beyond only expressing fluorescent proteins, we are actively testing whether our lead enhancers can also drive gene replacement for various monogenic diseases. While many neurological diseases are specific to a single cell type, certain diseases affect more than one cell type. We have found several enhancers that naturally co-express in multiple cell types, which is especially true for cell types of the same subclass, such as different types of inhibitory neurons. It has been more challenging, however, to find enhancers that co-express in two lineages that are more distant, like neurons and glia. We have recently tested whether multiple small enhancers can be combined into a single AAV to grant dual specificity. In this example, we added together two enhancers, one selected for astrocyte expression and another specific to inhibitory neurons. Due to the modular nature of these enhancers, we found we could now obtain dual specific expression in both astrocytes and inhibitory neurons. On the left, most cells expressing the fluorescent protein SYFP2 display the characteristic astrocyte morphology. But when we co-stain for the inhibitory neuron marker GAD1, we found about 25% of the total label cells were inhibitory neurons. We hope this dual specificity enhancer will prove valuable for gene therapy applications that require expression of therapeutic cargoes in both astrocytes and inhibitory neurons. Up next, you'll hear about some exciting work from Natalie Weed on the enhancer discovery in the striatum. Thank you for your attention.
To conclude our team talk, I'm going to be discussing subclass enhancer-driven AAV expression in striatum. As Brian mentioned, the striatum is a major area of interest in gene therapy. It consists of a mixed population of cells, including medium spiny neurons, large cholinergic interneurons, and other interneurons. Medium spiny neurons can be further divided by their associated circuits into indirect and direct pathway MSNs. The indirect pathway is characterized by axon projections to globus pallidus externa and is associated with the inhibition of movement, while the direct pathway is characterized by axon projections to substantia nigra pars reticula and is associated with the generation of movement. Therefore, the striatum is an area of high interest to movement disorders. Using internally and externally generated ATAC-seq datasets, we rationally selected putative enhancers based on our area of interest, in this case, striatum circuit enhancers. Our strategy includes selecting enhancers, generating AAVs, and delivering via retroorbital injection in mice, followed by imaging of native fluorescence. We have found putative enhancers that target pan-MSN circuits, direct MSN circuits, indirect MSN circuits, and locally projecting large cholinergic interneurons, as shown here. While we're able to conduct brain-wide screening in mice via retroorbital injection, we also want to have an equivalent in primate. There's no clear proxy for brain-wide expression specificity exploration in monkeys, as systemic delivery is inefficient and intraparenchymal injection is costly. So we have decided to use an ex vivo slice culture and virus transduction platform. This ex vivo platform involves collecting and subsectioning tissue to maximize enhancer screening within a given tissue. As previously mentioned, we have found strong labeling in mouse RO that's specific to indirect pathway labeling, as shown here in native fluorescence in green. From here, we proceeded to screen our enhancer in an ex vivo primate slice culture, in this case in putamen, where we can see robust viral labeling in green. These cells show MSN-like morphology, and additionally, the large cholinergic-like interneurons, as seen here in red, are excluded from this labeling. After this positive screen result, we moved to in vivo stereotaxic injections utilizing the same scheme John shared with us before. From here, we collected the stereotaxically injected macaque putamen and performed IHC. Here in red, we can see anti-DARP32, which is a pan-MSN antibody that shows widespread and uniform labeling of MSNs. Furthermore, we can see anti-MTFP1, which is a reporter protein from our indirect circuit AAV enhancer, in green that shows MSN-like morphology labeling with non-uniform distribution and incompleteness. As indirect MSNs are a subpopulation of MSNs, this is actually a very promising result. However, further MFISH validation is required to determine if these are indeed direct or indirect pathway. In conclusion, we have generated a complete set of MSN enhancers in mouse striatum circuits, including pan, direct, and indirect medium spiny neurons, as well as cholinergic interneurons. We're working to confirm our enhancer activity is conserved between mouse and macaque using MFISH technology. And additionally, we're exploring other subcortical brain regions and cell types. We're continuously refining our enhancer specificity. As we generate single-cell ATAC-seq data, as discussed by Yi, we hope to move from subclass to cell type specific enhancer discovery. Together, this work, along with other efforts throughout the Institute and beyond, we hope to further our goal of advancing research and developing potential gene therapy applications. Thank you.
So we'd like to end today by thanking this really large group of people who have been critical for making this project happen. First and foremost, we want to thank everybody who's involved in the Human Genetic Tools team, especially Jonathan Ting, who's not presenting today, but co-leads this project. The Mouse Genetic Tools team, who we work very closely with, all the members of the CNS Gene Therapy project team. There's also a lot of work done by tissue processing, the molecular biology team, the viral technology team that's packaged all of our viruses, as well as the human IVSCC team who's been testing some of these in slice culture. We've had fantastic support from the TCM, ACT, and LAS groups that maintain all of our animals. And we'd last like to thank some really, really great collaborators. And I'd specifically like to call out Greg Horowitz, who's really enabled us to do in vivo primate research. Last, I'd like to thank Adeline, Hanqui, Christoph, John, Todd, and Alan for really great leadership. So with that, thank you for your attention, and we'd be happy to take questions.
Okay, thank you everyone for joining us for day one of our Showcase Symposium and for your questions. This concludes the public webinar portion of our event for today. Attendees are welcome to visit the Allen BrainMap Community Forum to submit a question for this and all previous project talks. We have dropped the link in the chat for your convenience. I'd like to thank the EM group, the Visual Behavior group, and the Human Cell Types group for their talks and all the work they put into those. And I'd like to thank our NGL speakers for the day, Bianca and Lucas, and we look forward to the next 3 NGL presentations tomorrow, 2 more project talks, and as always, we also wish to thank our founder, Paul G. Allen, for his vision, encouragement, and support. Allen Institute employees and NGLs will see you in the Q&A sessions and roundtable discussions. Please leave this webinar session and join those via separate Zoom links in your calendar invites.